So I wanted to welcome everyone to the Nicholas Vogelzang Symposium today. This session, we're going to talk about microbiome as a new frontier in SWOG clinical trials. So really the new applicability of the microbiome. I'm sure everyone's heard about it. We have a great panel discussions coming up today. But I really first wanted to talk a little bit about Nick and the first experience I had with SWOG. So I was speaking with Ian Thompson. He had brought me and was encouraging me to get involved. And after the first U session on Friday, I was asking him, well, who do I need to talk about to get more involved? And Ian said, oh, I see Nick over there. Let's go have a beer. So I got to have a beer, and then a couple of minutes later, Ian left, and it was just Nick and I, and we sat there. He sat with me for almost 45 minutes talking about career. We talked a little bit about microbiome even then, and he said if it has anything to do with the immune system, latch onto it and don't let go. So it was a really nice meeting with him, and just kind of speaks to his mentorship and a desire to have the younger generation come up and take leadership positions. So I was really thankful for him for that. And Monty sent me some slides as well, and some pictures of how his mentorship has changed the lives of probably many of the people in this room and in the other sessions. He just kind of reached the entire bit of SWOG. So thanks to Nick. I wanted to bring this up because we'll be talking about the microbiome. And I think a long time ago, even Hippocrates said all disease begins in the gut. Now, microbiome doesn't all have gut-related. We can check the urine. It could be on skin. It can be anywhere. So how does this relate to other diseases? And how do we get away from just the old culture techniques that was invented in 1882? This is actually Microbia. It's a microbiome museum in Amsterdam. Of course, I went to that. So once we start sequencing it, we go back to 4th grade biology when we talk about the biomes, and the desert doesn't have the same animals in it as the rainforests. And so this is kind of from this work, this Nature paper, a lot of this work had foundationally became. So how does this work? So essentially what you eat and your exposures and your microbiome can all relate into the gut. And we're going to have great discussions about that today. And this is our lineup. We'll try to keep on time, but I wanted to ask Mani, did you want to say anything about Nick? Okay, so we will go right to Dr. Rowdy. He's our guest speaker and keynote speaker today. And I'll get his slides pulled up.
While Bertrand's coming up to the microphone, I'll just add that, Dr. Rudy is really one of the foremost thought leaders in the microbiome. I think many of us, whenever we write a paper on the microbiome, start with 3 pivotal citations related to immune checkpoint inhibitors and response based on the microbiome. And Bertrand is first author on one of those key science papers that came out in 2017. And since then, he's really done transformative work to bring forward therapeutic clinical trials. Bertrand is based at the University of Montreal. He and I are embarking on a number of, I think, really exciting collaborations. And he's actually going to be sharing today a lot of confidential data. So slides that are marked "do not share," if you don't mind just respecting that as he goes through his presentation. Thanks a lot, Dr. Rudy, for joining us.
So thank you, Monty, for this warm welcoming. And it's a great pleasure for a Canadian young PI to be a speaker at the SWOG. So today I'll give you a brief overview of the gut microbiome, including 2 unpublished data from my lab. My conflict of interest. I put these slides for you to remind you that back in 2015, the gut microbiome was still in its infancy. Nevertheless, in less than a decade, 4 major discovery propelled the microbiome from a joke, as my nickname, Mr. Caca, to now one of the hallmarks of cancer. So what happened over the last 10 years? So first of all, the microbiome as a biomarker of immunotherapy response. So during my PhD, we performed metagenomic sequencing of 100 patients, mostly with non-small cell lung cancer. And we discovered that one bacteria, a commensal bacteria, so a non-pathogenic bacteria, Akkermansia muciniphylla was enriched in responding patients. We recently extended this observation in 338 non-small cell lung cancer patients, where we demonstrated that patients harboring Akkermansia muciniphylla had a 10% increase objective response rate compared to Akkermansia negative patients. Next, in order to uncover the intratumoral transcriptomic differences driven by Akkermansia, we performed RNA sequencing on the tumor of 50 patients and separated based on their microbiome presence or absence of Akkermansia. And you can see here on this heat map that patients positive for Akkermansia had more intratumor T cells and increased Th1 as well as interferon gamma signature in their tumor, meaning that if you had Akkermansia in your feces, you had a more inflammatory, distant lung cancer tumor. Of course, several groups around the world, including the group from Jennifer Wargo, Tom Gajewski, Nicolas Segata, performed similar analysis and metagenomic sequencing. And we just published or got this paper accepted 2 days ago in Nature Review, where we perform a meta-analysis of all the metagenomic data available online. Unfortunately, for you in this field, we were not able to find a large cohort of renal cell carcinoma patients with metagenomic data available online. Nevertheless, for lung and melanoma, what's striking is on the upper portion of the graph, you see these are the commonalities, so the red bacteria or bacteria that are increased in all the trials that are represented at the bottom, a bit small. And at the bottom, this part is the bacteria increase in non-responder patients. So I summarize here for you, but what's interesting is regardless of the country, the tumor, even the type of immunotherapy, we have now defined a group of beneficial bacteria. And for you to remember today, so Ruminococcus, Faecalibacterium, Akkermansia, Eubacterium are known to be beneficial bacteria. Conversely, we have now harmful bacteria that are known to be resistant to immunotherapy, such as Clostridium, Enterobacteria, and Ungatella. The second major pillar discovery was that antibiotics block immunotherapy efficacy. Indeed, in 2018, while I was doing experiments in mice, we were giving broad spectrum antibiotic to kill pathogen, but also commensal bacteria. We realized that immunotherapy was also inhibited. We went to see our friend Matt Edman at the Memorial Sloan Kettering, and we also realized that patients with non-small cell lung cancer that took antibiotic 1 month before immunotherapy initiation had a shorter overall survival. This provocative experiment has actually been replicated, and that's a brand new figure from a paper that's coming out soon in more than 100 publications, and 43,000 patients around the world were tested for the deleterious impact of antibiotic. And you see that the hazard ratio is 1.71. So clearly, antibiotic for patients treated with IO chemo, and again, to the best of my knowledge, and for you guys, there's no IO TKI data on antibiotic available currently. So what do we know about antibiotic-related dysbiosis? So we published 2 papers, one with our Japanese collaborator, demonstrating that antibiotic, as expected, decrease the global diversity of the microbiome. Interestingly, and now you're expert in the microbiome, in red, you see that these are the bacteria that were decreased by patients receiving antibiotic, and they lose Ruminococcus, and I told you Ruminococcus was a good bacteria. Conversely, patients that receive antibiotic had an upregulation of Ungatella, a deleterious bacteria. Interestingly, and still we have difficulty to understand that, we performed the same experiments, this time in kidney cancer from France, and we found exactly the same antibiotic-related signature. So what do we do? So first of all, as clinicians, we have to judiciously prescribe antibiotic and make sure that patients really need antibiotic. However, as you know, some patients have clearly documented infection. So what do? So we joined force 2 years ago with a French company called Davolterra, or developed a charcoal capsules that opens up in the colon. And the hypothesis was that we would be able to absorb the antibiotic in the colon and prevent antibiotic-related dysbiosis. Therefore, we launched a clinical trial, a randomized trial of 70 healthy volunteers. The healthy volunteers were randomized to receive either the charcoal capsule alone, either ceftazidime avibactam IV or ceftazidime avibactam plus the charcoal capsules. And the term arm received piperacillin tazobactam alone or piperacillin tazobactam plus the charcoal. First, we looked at the plasma concentration of the antibiotic to make sure that we did not decrease the beneficial effect of the antibiotic, and you see the plasma concentration was maintained. Conversely, when we look at the concentration of the antibiotic in the feces, you see that in the ceftazidime avibactam group, those that received the charcoal capsules completely inhibited, or the charcoal was able to absorb the antibiotic in the feces. We then look at the microbiome diversity, and you first see that in red is the patient that received antibiotic alone. So they receive antibiotic for 5 days. At 6 days, we have the nadir in the lowest rate of bacterial diversity. However, for us and for me as a clinician, you can see that even 37 days after 5 days of antibiotic, the microbiome was not fully back to its initial composition. Interestingly, patients or healthy volunteers that received the antibiotic plus the capsules had a small decrease in their diversity, but rapidly within 10 days, it went back to normal. We, of course, look at the metagenomic data of this healthy volunteer, and you see on the left, when you compare to the baseline, here we don't see what, the pointer does not work, but when you see on the baseline on the left at day 1, all the greens are the bacteria. At day 6, the second column, you see that everything is black. That means we lost all these bacteria. And on day 16, some of these bacteria are back. Conversely, in the other arm, the patient that received piptazo plus the charcoal capsule, on day 1, you have a nice diversity. Day 6, we lose some bacteria, but clearly maintain more than the other arm. And on day 16, we're also back to baseline. Interestingly, when you see the bacteria that were preserved in the healthy volunteer that received the charcoal capsules, Alistipes, Bifidobacterium, Eubacterium, and Ruminococcus. As a proof of principle, we then performed fecal microbiota transplantation using the healthy volunteer feces, thank you, in germ-free mice. And so we took the feces of these healthy volunteers at day 1, before they receive antibiotic. We perform oral gavage in mice. We wait for 14 days. We put the tumor and we give PD-1. So at day 1, when we took the feces of the healthy volunteer, so this is a pooled experiment of 3 healthy volunteers, on the antibiotic group, their feces was able to maintain PD-1 activity. Same thing for the other group. However, at day 6, so after 5 days of antibiotic, when we took the feces of these healthy volunteers at the same time, you see that we block PD-1 efficacy, so potentially the patient would have not responded to immunotherapy. However, when we took the feces of the patient that received antibiotic plus the charcoal, their feces were able to maintain anti-PD-1 response. Very rapidly, we then, of course, look at surrogate marker, and in the mice, there was upregulation of CD8 over Treg in the mice that received ceftazidime plus ADAV feces. So that's quite interesting. So the third major pillar was, how can we modify microbiome in murine experiment? So we first demonstrated that if you take feces from non-responder, you humanize a germ-free mice. So you transfer their feces in a germ-free mice, you put a tumor, you give immunotherapy. In approximately 80 to 85%, you can actually predict the outcome of the patient. And this has been replicated by Wargo, Gajewski, and recently in a nice Nature paper. And you see that the non-responder, the feces block anti-PD-1 activity, responder feces work. That was nice, but of course, as oncologists, we want to increase activity. So we first thought that, oh, we could do a salvage FMT using feces of renal cell carcinoma patient in complete response. So this is what we did, again, only in mice. We took feces from 8 renal cell carcinoma resistant to immunotherapy, transplant them in mice, put a RANCA orthotopic kidney tumor, give the mice PD-1 CTLA-4. If you only do that, there is no activity. However, if at the time of PD-1 CTLA-4, you do a FMT from a patient in complete response, in mice, again, we were able to restore double immunotherapy efficacy. We did a similar experiment, this time in non-small cell lung cancer. We took Akkermansia negative patients, performed FMT in the mice, and we performed oral supplementation with Akkermansia, and we were able to restore anti-PD-1 response. While we were doing this in the mice, in Israel and in Pittsburgh, they did this in patients with metastatic melanoma, refractory to immunotherapy, FMT from complete responder patients in refractory melanoma patients. And 1 out of 3 patients that was re-challenged with immunotherapy started to respond again. So then when I moved to Canada in 2018, we were aware of these data. So we decided to go first line, and we completed the first Canadian trial combining FMT, this time of healthy volunteer, in naive anti-PD-1 metastatic melanoma patients. And in Canada, we use oral capsules for the fecal transplants. So out of the 20 patients enrolled in this trial, they received FMT plus anti-PD-1. You see that there is no toxicity of the FMT alone. When we look at the IO plus the FMT, we had five patients develop grade 3 toxicity, which is what we expected in the single agent treatment. However, when you look at the waterfall plot, so if you're not a melanoma expert, the randomized trial predict that objective response rate of 45 to 44%. And in our trial, we had 65% of objective response rates with 4 patients actually in complete response. What's interesting is that there was no donor benefits, or the 3 donors led to either resistance or response in patients. We then look at the microbiome composition and evolution of the patients on the trial. And what's interesting, so this is a Bray-Curtis dissimilarity. So if it goes down, that means you become like your donor. If it goes up, you become like yourself, or you go back to yourself. So you see that if we separated non-responder and the responder before FMT, so everybody starts at 0, then 1 week after the FMT, everybody becomes like their donor. Nevertheless, 1 month and 3 months for the non-responder, they become like themselves again. Conversely, the non-responder, 1 week, they become like the donor, and this change is sustained over time. So there was possibly engraftment. We then pulled the data from Davar and Baruch paper, and they actually found similar results, that responder global microbiome was shifting towards the donor over time and was sustained compared to non-responder. Then we look at the microbiome composition. So this is a heat map of the on left is the feces before the FMT, 1 month after the FMT. And you see that the responder patient, they lost the enterocluster. And I told you initially that enterocluster was our worst enemy. Conversely, there was an upregulation of good bacteria, such as Eubacterium, Dorea, and Ruminococcus. Rapidly, we performed flow cytometry, and there was a nice upregulation of CD8 ICOS 1 month after FMT in responder. And then once again, we went back to the mice, and we performed oral gavage of the feces of the patients before they got their FMT, of 3 responder and 1 non-responder patients. And you see in these experiments, none of the feces were able to restore anti-PD-1 activity. Therefore, potentially, these patients, as I showed you initially, were doomed to not respond. Nevertheless, after the FMT, the 3 responder patients, when we transplanted their FMT, immunotherapy worked again. So potentially, because of our intervention, it's not a randomized trial, we showed that responder start to respond after the FMT. We looked at the microbiome of these mice, and once again, we showed that the same beneficial bacteria were increased in the feces of the mice only after they get the feces post-FMT transplant. And lastly, so another strategy to shift the microbiome composition is to use prebiotics. So we isolated from a Brazilian berry, a castalagen, a polyphenol polyphenol agent, sorry. And in germ-free mice, so without bacteria, we show that this polyphenol has no anti-tumor activity. We perform our favorite experiments where we took feces of non-responder. If we add the polyphenol, and the polyphenol with anti-PD-1, we see that the immunotherapy, there's a synergetic effect. And when we look at the microbiome of the mice that receive this polyphenol, there is an upregulation of, guess what? Ruminococcus, Akkermansia, and Alistipes. We then uncover a part of the mechanism using fluorescein-bound castellagent, our polyphenol. Through flow cytometry, we show that it preferentially binds to Ruminococcus, and we also perform a microscopy experiment, and we actually show that this polyphenol binds the surface of the bacteria and does not enter into the bacteria, and we're now actively working on the mechanism. In parallel, and thanks to these results, we launched in Montreal a trial combining Camus to PD-1 in melanoma and chemo IO plus Camus non-small cell lung cancer. And I hope that Montipal will tell you about his exciting Camus trial later today. So in conclusion, what have we learned since 2015? So I'm happy to tell you that finally the gut microbiome is now part of the hallmarks of cancer. We have signals of potential mechanism. Very nice paper in Nature last week on PDL2. The gut microbiome clearly represents a novel biomarker of ICI response. I've not talked about immune-related toxicity. Antibiotic blunt anti-PD-1 efficacy, we decrease diversity, we lose the good bacteria. Transfer of the feces of patients in mice represents a strategy to understand, but also to do different tests, to test new probiotics, prebiotics in murine experiments, because these experiments in mice at least work. In patients, we have now very good signal in melanoma for FMT. There will be new data presented at ASCO in 2-3 weeks. And lastly, with regard to probiotics, so as you know, Ceres is now FDA approved for the treatment of C. difficile. You will hear more about a probiotic positive trial in oncology. Nevertheless, what we need to discover together over the next 5 to 10 years. So first of all, the mechanism, it's still very early. With regard to biomarker, I showed you a meta-analysis, but really, is it one size fits all? Is it the same bacteria going to be good for every cancer, every IO combination, every country? Do we know, is there a real cancer-related dysbiosis? With regard to FMT, so what is the best source of the donor? We use healthy volunteer in Canada because it's forbidden to give FMT from cancer patient in Canada. Other trials use patient in complete response. How do you prepare your patients? You do GI prep. Us, we give Golightly the day before the FMT. The Israeli group used antibiotic. Do we need it? What is the best preparation? How many FMT do you re-challenge FMT? But probably the more important or scientifically driven strategy would be to show that if there is engraftment, you don't do anything. But if there's no engraftment, then you can do another FMT from another donor. However, and this again for us in the clinic, is we also need to develop tools to have rapid microbiome composition, because if you collect feces in a trial, you do metagenomic, the bioinformation results, you get the result a year later. So this is definitely not good for the clinic. So we have to find new way to have a rapid tool and kit to do this. With regard to probiotics, you'll hear more about it today. But here the big question is, do we need live pasteurized bacteria? Is it one bacteria enough, a consortium of bacteria? You'll hear, of course, probably only the good news, but there are some consortium bacteria that the trials are negative. Why? And we have to understand that. And in terms of clinical design, and this is what we will, I guess, discuss after, what is the good endpoint? What is the good microbiome primary endpoint? In a clinical trial, should non-dysbiotic cancer patient be enrolled in your trial? And how do you randomize? And lastly, I hope we'll be able to work all together in a nice collaboration to answer all these questions. So of course, I want to thank my team, Myriam, who's running the lab, Ariel Elkrieff, who's running the clinical trial, and my mentor, Laurence Vogel. Thank you very much.
Spectacular. And what we're going to do is we're going to hold questions until after Dr. Jami's talk. You've set the bar so high, Dr. Rudy, in so many ways. And first of all, the work that you've done is just stellar in such a short period of time. And I'll also say beyond that, you managed to actually finish your talk 2 minutes ahead of the allotted time, which is incredible. I think that Nick would be so proud to see that it's standing room only in this room, but I will point out that, we do have a couple of open seats on the left-hand side of the room here, in case. Oh, perfect. Oh, thank you, Joe. That's awesome. But I'm really delighted to introduce Dr. Ajami, who's actually the executive director for scientific research within Prime TR, which is the MD Anderson machine. It really is an incredible machine that's really putting out amazing, amazing work in the microbiome space. Dr. Ajami, as I'd mentioned, is a part of the MD Anderson group that really is one of that triumvirate of papers that established the relationship between checkpoint inhibitor response in the microbiome. And since then, just as Dr. Rudy's done, he's really taken it many, many steps further into trials evaluating diet and involving other interventions. But we've asked him specifically today to comment on the role of diet in the microbiome. Dr. Ajami, welcome.
Thank you very much, Monty and Liz, for the invitation. And we're putting together an incredible panel. And this is not planned. I know, Bertrand. Disclosures first. Perhaps the most important is that I'm not an oncologist. I'm a microbiologist by training with a PhD in virology, but I've been in the microbiome field since probably 2010 and the early days of the Human Microbiome Project. Again, not planned, but you'll see that my opening slide looks a lot like Bertrand's final slide. So we usually hear this, right? Every microbiome talk talks about how we have a lot more microbes and genes than human cells and whatnot. And this slide usually, follows with the statement that what we think is that the microbiome is 99% modifiable. And this is what keeps me up at night, really. These are the questions that we continuously ask ourselves in our group. What factors shape our microbiomes? Is it genetics? Is it environmental factors? How are healthy patients different from cancer patients when we look at modifiable factors? How and when to best profile the microbiota, so the cells or the microbiome? Are we leaving a lot out of the table because we're only looking at taxonomy, only looking at DNA-based profiling methods? Do we need to start looking at secreted products, at perhaps cellular products that we don't get through NGS? What are the windows of opportunity to intervene on the microbiome from birth to adulthood? Can all microbiomes be restored, or are there microbiomes that actually need to be replaced to get that phenotype of interest? Similarly, do all microbiomes respond the same way to the same factors? So if we give all of us the same diet in this room, do we all respond the same? We don't have trials large enough to try to answer these questions. When we give someone FMTs from responders or from healthy individuals, do they all engraft the same? Do we all have the same capacity to engraft, to take on those new bacteria and colonize, perhaps more than that, phage or eukaryotic viruses? How that interplay comes into play. Antibiotics, life-saving drugs, but they produce a lot of collateral damage, and we have now great evidence that supports the fact that they could be detrimental in the context of immunotherapy. So, given the fact that I'm at MD Anderson, we treat patients with a lot of these drugs. How do we put everything in context to offer our patients better opportunities to respond to the therapies that they're getting? How durable are these changes when we give someone FMTs? Bertrand just showed us beautiful data. His x-axis had 70 days out, and looking at changes after FMT. Is 70 days enough? Do we need to be engrafted, quote unquote, for years? Are there windows of opportunities? Do we need that engraftment to happen right before PD-1? So these are questions that we have yet to answer. And then how do the host respond? We look at things through the microbiome lens, or at least I do a lot. So I think the field is shifting towards more of a holistic approach. Liz told us earlier that he got into this field by grabbing into immunology, and I think that's the great amplifier. These microbes are immunostimulant. They modulate immune responses, and we oftentimes leave that out in our analysis. We love to show microbiome panel A, immune panel B, and we often forget because probably it's very difficult to put those two together and find signals going in both directions and how those interact. And then how can we translate this across diseases? If a dysbiotic patient with C. diff gets an FMT, does better, is that the same? Is that concept translatable to a cancer patient that is getting FMT, who previously progressed on anti-PD-1 or anti-CTLA-4 and now getting FMT for a better chance to respond to those drugs? Can we translate those concepts across disease states? And these are all questions that keep me up at night because I am tasked at MD Anderson of creating a group and building a group equipped to respond to all these questions, right? So when I think about these questions, I go back to microbiology, microbial ecology, and I think about, again, what shapes our microbiome, how is microbiome developed? Try to look for answers within those ecologies. So we know that there are different developmental phases of the microbiome from birth. Some even propose that there's a microbiome pre-birth. Not a lot of evidence there or good evidence, but we find microbes at every place we look. And we find microbes from very early in life, all the way through adulthood. So we know there are phases. So we expect some dynamics of these microbiomes. We know that there are unique niches in the body, right? If you look at the oral cavity, you can find different microbiomes from a single person in different places of the oral cavity at the same time. And that just is an incredible diversity. That makes everything that we do in the lab a lot harder because there's not a consensus. There's lack of uniformity in the things that we look. But nonetheless, biology has been super smart over millions of years, and we have functional redundancy. Despite the taxonomic and compositional differences of the microbiome across our body, we see that these microbes perform housekeeping functions that need to be there. So my counterpart in Japan, giving the same talk to another cancer group, otherwise healthy, has a very different microbiome than I do. And we got to understand that diversity and that heterogeneity when we plan studies, when we look at cohorts, when we plan interventions, right? We know now that environmental factors win over genetic factors in terms of how the microbiomes are shaped, right? We know that genetics play a big role in diseases, but that does not explain the total variability in the microbiome side. And another concept that is really rising up in our field, which is really not new. It's new for the microbiome researchers, but really not new for microbial ecologists or ecologists in general, is that we love to single out bacteria. We love to run our stats and we love to rank by p-values and by differential abundance analysis. And I'm guilty. We did a lot of this. But these are really ecologies. If you talk to an ecologist or a microbial ecologist, a microbiologist, which, by the way, we need more in the cancer field, they'll tell you that there are hierarchies. If bug A is producing a metabolite or a product B, that product B is taken by bug C and retransforming it into D, and then it's a whole change. So if you affect something up here, you need to expect it to alter the community below. So this keeps me up, again, as I said, and to try to answer all these questions, we formed PrimeTR. This is really the brainchild of Jennifer Wargo, a wonderful surgeon scientist that treats patients with advanced melanoma at MD Anderson, a pioneer in the microbiome oncology field. And we've come together to try to build tools, which is one of the things that I love to do the most, build tools and workflows and resources to try to unlock like new corners of science. And our team really supports the MD Anderson mission by coming up with what I think is exceptional translational research and to evaluate potential applications of the microbiome in cancer patients. So we're not a core. I'd like to open with that. We consider PrimeTR as a boutique shop. We come in together. We have an internal advisory board and an external advisory board made by subject matter experts when projects are pitched, and we put all our brains together and try to build sort of the framework for analysis for a project. And that could be designing a new trial, or that could be analyzing a data set. So we like to operate across the spin wheel of the microbiome research. And we do a lot of profiling. This is the foundational work. There's a lot of data out there, a lot of noise as well. We do some research and development to complement all of that. And then very interestingly, we do a lot of microbiome modulation trials, which is going to be the second part of my talk if I have time. So a lot of different profiling, very flexible with defined workflows to look at all types of samples beyond stool. We collect samples in many, many different ways, and we preserve them in many, many different ways to enable culturing, to enable RNA-seq analysis, to enable single cell analysis, and so on and so forth. We also do a lot of data mining at a place like MD Anderson, where it's a really data-rich institution and computational heavy resources are available. We do a lot of data mining to look for signatures among the noise of data, because this data that is produced not for microbiome purposes is usually very, very dirty. We've defined tools to look for viruses, for bacteria, to assemble genomes, to look for fungi, and we interrogate existing data sets to see if we can find some hypothesis generating information. Very recently, we started mining the TCGA data set, including exome and RNA-seq and whole genome shotgun for viral burden with a tool that we developed. And we're finding very interesting, I'm kind of calling them signals right now, not signatures yet. So these are all things or tools that we create to try to move the field further. A very exciting area of research that we are developing is tissue microbiome profiling. Another great resource at MD Anderson. It's a sample-rich institution, so we have a big banks of tissues. We want to understand what is the role of microbes within tumors, and we're developing that in collaboration with NanoString, following the great work by Matt Meyerson and Susan Bullman and others, and Ravid Straussman, as I'm pointing out here, where they identify microbes present within tumors. And what we don't know yet is what are the roles of those microbes in those tumors, where do they come from? Are they the same microbes that are coming from the oral cavity or the gut, et cetera? So these are all sort of the underlying questions behind the tools that we are generating. Very much needed in the field. Again, what I talked about in the opening slides is how do we integrate a lot of the data? Right? We consider this data to be independent vectors when they're really not. They're really interconnected. It's an extremely hard challenge. But I think, we are now, with the amount of data we're generating, we have a good crack, or a good opportunity to crack this one. One of the exemplary projects that we launched at MD Anderson is this project called Patient Mosaic. And the goal is to learn from every patient. Heterogeneity of cancers, heterogeneity of treatments, heterogeneity of responses. Some people respond, some do not. Some respond, but only after seeing toxicity. Others respond without toxicity. So how do we explain all that heterogeneity? And we think that the microbiome can be one of the pieces of that puzzle. Definitely not the entire answer, but could be one of those pieces. And the lofty goals for this project is to enroll 10,000 patients within 3 years and really profile as much as we can. So germline sequencing, tumor sequencing, microbiome profiling, you name it. All right, so for the second part of my talk, if I have time, I want to give you some insights on microbiome modulation trials at MD Anderson, which is a part that really excites me. We have done a lot of foundational data, a lot of cohort discovery, a lot of profiling, thousands upon thousands of samples. But I think that it's time to start testing some of these hypotheses in humans and with our patients to try to give us the opportunity to develop better strategies to treat them. So we have our platform is supporting a few modulation trials, including FMT for colorectal cancer, pancreatic cancer, FMT for IO colitis, but also diet. Justin Gregg and Jen McQuaid have ongoing dietary interventional trials for prostate cancer and melanoma, respectively. Advantages: can we use food as a drug? Is it possible? If we look at FMT, probiotics, prebiotics, I would throw in here antibiotics eventually. Can we use diet as a drug? Is it possible? So I'm going to share with you some of the early data out of the phase 1 of this trial. So phase 1 is in 10 patients of melanoma where there's no evidence of disease, so we can call them survivors. We have a professional chef at MD Anderson cooking meals for these patients, N equals 10, worth of 6 weeks. So every food that these participants are getting are cooked by a kitchen. We control their dietary intake, we control their calorie intake, we control what they eat. So the goal of this one was to ramp up this 10 people to 50 grams of fiber per day. And I'll tell you what happened. So exceptional compliance, very well tolerated. So we're good. We can eat beans, we can eat legumes, we can eat fruit. It's great. Just be mindful of the timeline. So baseline, 6 weeks of intervention, and then the last sample at week 12. So what do you think happened? At the baseline, we don't eat enough fiber. That's one of the things. Texans or Houstonians, these people around our institution, we don't eat a lot of fiber despite having a big Hispanic population, and we all love beans, right? So the good news is that over 6 weeks, everyone ramped up their fiber intake with this diet. So it's possible. Great. If I feed you for free, you'll eat. Awesome. It looks like grad students, right? You can bring pizza, you have a very well-populated journal club, but we're all humans. And look at this, 6 weeks post high fiber diet intervention, boom, we forgot. So what happens here? We got to get a lot smarter, a lot more creative, because, this is a good step one, but this is not something that is going to be very, very feasible across the board. So just have this in mind as we think about the new set of trials, about thinking about diet. In my opinion, diet is not enough. You need diet and a good microbiome. In my opinion, when you give someone an FMT, you need to sustain that FMT. How you do that? By feeding that microbiome the right things. Do we know what are the right things? That's another question. So the microbiome data follows up sort of that inverted V shape, where we see diversity and structural changes following those baselines, but at the endpoint, right, post-intervention, we see that these participants reverted back to their microbiomes. The good news, we can instill changes in the microbiome in a short period of time, and that's exceptional, right? If we do this pre-PD-1 or pre-immunotherapy, we can probably have a good window there to achieve the responses that we want. We can go from a Fecalibacterium not having a lot of friends to play with in the gut to having a lot of friends, a lot more connections, at least statistically derived on correlations. We know that these bugs or this bug, which is a fiber responsive bug, can improve the network strategy. Structurally, the microbiome also changes. We see that those participants that started with very low fiber and ramped up to very high fiber, those had the biggest changes. I'm using sort of the same metric here that Bertrand used, Bray Curtis, to measure the distance between baseline and intervention. So this is all really, really great news. We can modify a microbiome in a very rapid way through diet. So I think that's still very positive. We looked at metabolites in serum, and we can also see that these participants sort of shift in their profile of metabolites during the intervention. But unfortunately, like we saw with fiber intake, like we saw with taxonomic and structural changes in the microbiome, we also see that they revert back to their baselines, unfortunately. So that tells us about durability. So we're excited about the phase 2 part of this trial. It's a bigger N, it has bigger endpoints. We're not gonna be able to correlate this with response yet. I think we need hundreds, multi-institutional cohorts to really crack this one, but this is where you start. And I'm glad that at a cancer institution, we're giving ourselves the opportunity to look at how we do this through the microbiome. So the key takeaways: microbiomes are responsive to environmental changes, including dietary interventions. It's extremely hard. You need a professional chef in a kitchen cooking meals for everyone for 6 weeks, but it can be done, right? There are other examples in our field in cancer were extremely hard achievements, but they're possible. Multi-omic profiling, we need to be thinking more about this. We need the experts in the world not to be developing the next Candy Crush, but to develop really how do we integrate all this data together, right? So we got to be competitive and lure those people to our field and to Texas, which is hard. Heterogeneity in baselines and heterogeneity across the room makes our world very, very difficult. But again, we got to test different interventions to see if we can create these funnels where we drive our recipients or our patients to the optimal microbiomes, whatever we decide those are. And test FMT, test antibiotics, more precision-based tools, diet, etc. So I'll leave you with that. You can read this, and I really acknowledge patients and their families, our funding programs, which is the Moonshots programs, our donor foundations, and collaborators, and happy to take questions in the panel. I hope I was sort of on time.
Okay. Yeah.
Awesome. Thank you.
We have a few minutes to take some questions.
Rick, you want to?
Rick?
I'd like to start. So I'm Rick Banks. I'm stepping down as the bladder cancer patient advocate. My replacement is back here. Please introduce yourself, Daryl Nakagawa. I thought this was a great presentation, and I'm particularly interested in something I didn't hear about, which is the impact of smoking on microbiome, because I had suspicions and Google confirmed them that there is. Remember, I'm one of the few people in the room that's not a doctor, so I had to use Google, and it looks like there's some implications there, which seem to me would have some clinical implications as well as some research implications. So I'd be interested in any comments on that.
Yes, so definitely smoking change your microbiome. But what's striking is when we compare microbiome of non-small cell lung cancer, and in North America, as you know, 80, if not 90% of our non-small cell population on IO are past or current smokers, to data from melanoma or even renal cell carcinoma. And what's even more striking is even if we compare them from France, Canada, UK, Japan, it's the same group of bacteria that are enriched in responder and the same group of bacteria that are decreased or upregulated in non-responder. So we still don't understand fully, and I think it goes down to the ecosystem, which ecosystem of a responder versus a non-responder. And then to answer also your question, so in the Netherlands, they have a beautiful program of microbiome sequencing, and their objective is that they have sequenced 10,000 healthy volunteers to predict who will develop cancer. Because my future dream is when we turn 40 or when I'll turn 40 or 50, is instead of doing colonoscopy, mammography, rectal exam, we just look at your microbiome and you say, oh, you have bacteria A, B, C. Oh, you're at high risk of colon cancer. You need colonoscopy 6 months. Whereas you have bacteria B, C, D, or you don't need any mammography, you're good. And so in the Netherlands, they, of course, have been pioneering this. And unfortunately, they have not been able to predict on 10,000 patients. They just were able to say those that will develop lung cancer. But then when we asked them, okay, but correct for the microbiome of smokers, it was the same thing. So their signature was not developing lung cancer, it was a signature of smoking patients. So this is the best way we can answer your important question. And same thing between male and female. There is definitely a difference, but in all trials, we never separate. Same thing for BMI and same thing for age.
If I could make a quick comment. So I do a lot of you'll hear a little bit later, in bladder cancer. And I don't have this slide specifically, but if you look at the specific microbiome of patients with bladder cancer, who we know a lot of them are smokers or have been smokers in the past. And then you look at the healthy cohorts, which we specifically picked to be non-smokers. Bladder cancer patients who are non-smokers typically cluster closer to the healthy population versus those who are actually smokers, particularly those that are current smokers. And now we know that their outcomes are much worse in terms of chemo and immunotherapy. Now, is it the microbiome that's driving those outcomes, or is it the smoking really itself in terms of the carcinogenesis and metabolism that's driving it, or is it just a vicious cycle of go back and forth? So there is significant clustering of those smoking patients versus non-smoking.
Rick, that was a great question. We actually have 57 people on the online audience. So as you approach the microphone, if you could just introduce yourself really quickly to them, that would be terrific, just as Rick did. Let me start here.
Hi, I'm Paramita Datta, a medical oncologist from San Antonio. Excellent presentation. My question is, can differences in microbiome explain differences in cancer in different parts of the world? And can FMT be used for cancer prevention in future?
So we know from mice that if you inoculate 3 different types of tumor, and that's a cancer discovery paper we published, Yonekura 2021, the microbiome of the mice will change because they have different tumors. So potentially, and again, we don't have enough data, and that's why Nadim says that we need to sequence more. And I think that potentially patients with cancer have a different microbiome than healthy volunteer. And then within the cancer-related dysbiosis, there's also a cancer histology difference. Is it driven by the smoking? Is it driven by the sex? Is it driven by the BMI? We still don't know. And then what about different country? That's another level of complexity, and we don't know yet. Can FMT cure cancer or prevent cancer? That's, of course, now my dream. But again, before giving this claim, we have to, first of all, be able to show that we can increase IO efficacy in the metastatic setting, then go neoadjuvant, and then we'll go to cancer prevention.
Thank you so much.
I'll say something real quick about that last point. So in colorectal cancer, there's a very strong signal of tumors having oral bacteria, Fusobacterium nucleatum, subtypes of species animalis. So strain diversity is extremely important. That's another thing that I didn't talk about. Parvimonas micra, oral bacteria. Those bacteria are really not supposed to be below our throats. Like really, they shouldn't be there. But if we find them in the intestinal tract, then that's a flag, and we should pay attention to that. Okay. And we treat that with an FMT, we don't know. Antibiotic, we don't know yet. But we know now enough about these ecologies where we can probably say that bug shouldn't be there.
So We'll do one more quick question because we're going to get going.
Yeah, hi, Amir Goldkorn from University of Southern California. Thank you for these amazing talks. So do we have any evidence in mice or in human subjects that FMT alone can alter the immune profile and perhaps induce or even sustain an immune response in the absence of checkpoint inhibitors?
Quickly, so in our FMT trial, we do FMT 1 week before IO, and there was one patient who got his FMT, and 3 days later called the physician. My lymph node is decreasing. The physician said, "I don't believe you. Come to the hospital," and the lymph node decreased.
Well,
companies don't like that. I like the story.
and you know, I had a sense of us having lots of great dialogue around this. So the last hour of the symposium is really dedicated to what I hope will be a really terrific Q&A session, and we'll discuss some pragmatic trial designs and really bring this back to the clinical, you know, as you've alluded to, Amir. It's my pleasure to introduce Dr. Nazli Disman. I wanted to say a couple of words about Nick in relation to Nazli. You know, I know many of us around the room know Nick is a terrific academic mentor. He certainly guided me and many others that I'm looking at here in the room in terms of our early careers and beyond. And I would always approach him for advice. Those of us that practice oncology in the Southwest have had also probably the pleasure of seeing second opinions and patients of Nick's. And it's just remarkable, the level of care that he delivered. I think we have some advocates in the room who would testify to that as well. But, you know, I used to really enjoy reading through his social history, which would include, you know, random details about the patient, their favorite flavor of ice cream, you know, what they used to, you know, sort of enjoy doing on weekends and so forth. And, you know, I really thought that that special touch is what made him not just an incredible mentor, but an incredible physician. Nazli actually came to work with me from Turkey about, it was 6-7 years ago now, at City of Hope. And the first thing I did was I got on the phone with Nick and I said, you know, will you take Nazli and a couple of my other fellows and have them spend a couple of weeks with you out there? So they did just that. And I think the experience was invaluable. Now Nazli is actually finishing up at Yale and starting her oncology fellowship at MD Anderson, where Nadim is. And she's going to tell us about the gut microbiome and RCC. Nazli?
I'd like to thank the organizers for the opportunity to talk about the gut microbiome directed strategies in renal cell carcinoma. My disclosures. There had been a tremendous progress in our understanding of the interactions.
Can we activate the microphone, the speaker microphone, please?
Is it working? Maybe I should be.
This one's working here. I don't know if we can swap this out.
Good morning, everyone. I don't know if you heard, but I thank the organizers for the opportunity to present here in my disclosures. And I was just saying that there had been a tremendous progress in our understanding of the interaction between the gut microbiome and outcomes in oncology. As Dr. Rowdy and Jami discussed already, among all these studies, a large number of them included patients with renal cell carcinoma. When we juxtapose the studies that included patients with renal cell carcinoma and looked at the clinically relevant species, we see that there are several species that appear to be associated with different outcomes, and some of them, such as Akkermansia mucinifera, are consistent among studies, but there is a significant heterogeneity that is evident both in VEGF-TKI-treated patients and immunotherapy-treated patients, perhaps highlighting the fact that we should be studying other aspects of microbiome than taxonomic composition and looking at the function, as Dr. Jami mentioned. As we understood this interaction, the further efforts are being dedicated to utilizing microbiome-directed interventions, FMT, dietary interventions, lifestyle interventions, live bacterial products, and probiotics are being studied in renal cell carcinoma as well. FMT experience in melanoma, as we've heard today, shows that FMT has an acceptable safety both as a monotherapy or in combination with different anti-cancer treatments. And it appears that it leads to enhancement of systemic immune response and response to treatment as a result of successful FMT engraftment. FMT studies in renal cell carcinoma were first published in 2020, looking at the impact of FMT on the treatment-related colitis with TKIs. Investigators enrolled patients with MRCC with tyrosine kinase inhibitor-induced diarrhea. Patients were randomized into either receiving healthy donor FMT or placebo FMT, and the healthy donor FMT achieved successful donor strain engraftment and resolution of symptoms as early as a week. There are other FMT studies ongoing in MRCC space. Dr. Maliki and colleagues in Canada are testing healthy donor FMT as a preventative means of immune-related colitis. The results of the first 10 patients was presented at ASCO in 2022 and showed 30% rate of immune-related colitis with first-line nivo-ipi in first 10 patients. Otherwise, FMT-related adverse events were mainly limited to gastrointestinal tract and were at grade 1 or 2. The recruitment for this study is ongoing. FMT appears to be a emerging therapeutic strategy in several different settings in oncology, and there are no safety concerns so far that we saw, but it still comes with some challenges associated with social acceptance, manufacturability, or cost. So a potential approach that could circumvent this could be the use of live biotherapeutics. For example, CBM-588 is a live bioproduct that contains Clostridium butyricum, which in preclinical studies showed that to increase lactate use and butyrate production in the gut lumen and increase abundances of the Bifidobacterium species and the levels of short-chain fatty acids in GI lumen. In a retrospective analysis in non-small cell lung cancer in Japan, patients who took CBM-588 supplements in addition to their prescribed immunotherapy had better objective response rate and prolonged progression-free and overall survival compared to those who did not take CBM-588. We designed a prospective phase 1 randomized study enrolling patients with metastatic RCC with clear cell and sarcomatous histology and intermediate or poor risk disease. Patients were randomized into either receiving nivo-ipi or nivo-ipi with CBM-588 in a 1-to-2 fashion. Primary endpoint of the study was the change in the abundance of Bifidobacterium species from baseline to week 12, and clinical outcomes and additional translational endpoints such as serum cytokine levels, circulating cell populations, and further studies of gut microbiome were secondary endpoints. In this study, we didn't meet our primary endpoint, and Bifidobacterium species remained stable from baseline to week 12 in both arms. But when we looked at the patients who received CBM-588 and responded to treatment, we saw an increase in the Bifidobacterium species, which may suggest that there is a certain patient population that achieves a gut microbiome modulation and has better benefit with the immunotherapy. When we look at the clinical outcomes, we've seen striking results. The PFS with nivo-ipi CBM was 12.7 months versus 2.5 months with nivo-ipi. Of course, I have to highlight that the study is very small sample size and was not powered to assess clinical outcomes. We also went ahead and analyzed the data after a long-term follow-up, and currently the median progression-free survival with nivo-ipi and CBM is around 36 months range. 58% of patients with CBM-588 had response to treatment, and versus 20% in nivolumab ipilimumab arm. And the outcomes appeared to be durable with CBM-588, as seen on this swimmer's plot in gray. In terms of adverse events, grade 3 or 4 adverse events were seen in 50% of the patients in the nivo-ipi arm versus 52% in nivo-ipi CBM arm. Although there were differences in terms of the type of the adverse events, the safety profile appeared to be comparable. We also did several translational analyses. When we looked at the circulating cytokine levels, we've seen significant increases in the circulating levels of chemokines such as CCL2, CCL4, CXCL9 or 10 in patients who received CBM-588, while these cytokines remain unchanged in the nivo-ipi arm by time. Also, regulatory T cells have increased in the nivo-EP arm by time, while they were unchanged in the nivo-EP CBM588 arm. There are other live biotherapeutics that are being studied. One of them is MRX-0518. This compound is a lyophilized formulation of Enterococcus gallinarum, which in preclinical studies were shown to be associated with enhanced immune response in a study of 15 patients with renal cell carcinoma who previously had disease progression on immunotherapies. MRX-0518 in combination with pembrolizumab led to durable and deep responses in a small proportion of patients. As Dr. Jami has mentioned, now we have more information regarding the association between fiber intake and the outcomes in melanoma. In renal cell carcinoma, we don't have a similar study, but what we know from the population-based studies is that low fiber intake is associated with high risk of renal cell carcinoma. So in renal cell carcinoma, dietary interventions are being tested in prospective settings, such as the one Dr. Jami mentioned is also enrolling patients with renal cell carcinoma. Another study is looking at ketogenic diet versus standard diet in patients with renal cell carcinoma in combination with nivo-EP. Our new study with first-line nivo-cabo plus or minus CBM588 will be an oral presentation by our colleague, Dr. Ibrahimi at ASCO, and FMT studies with healthy donor FMT in renal cell carcinoma ongoing, and then another study in Italy looking at responder FMT in patients who received pembro and axi. So for conclusion, it is evident from clinical and preclinical evidence is that microbiome-directed interventions will enter the later phases of the clinical studies. So there will be several considerations that needs to be really taught in detail. The studies in the past had patients with different stages or different settings, which needs to be homogenized. In addition to what we've discussed so far, I think I would say that the risk of immune-related adverse events need to be considered as well with immune related with microbiome directed interventions, and of course manufacturability, cost, ease of access for all patients are all important. As Dr. Paul mentioned, I was lucky to meet Dr. Vogelzang, so it was an honor to present here, and thank you for your attention.
I'll share with you that I'm really begging IT to keep your City of Hope email address active in your IT access. Stephanie Anderson. It's a real pleasure to introduce our next speaker, Dr. Mukavina, who's just a force of nature. I think many of you may know her in the urology community. She's currently finishing up in Fox Chase Cancer Center, and she's going to be joining faculty at Case, I believe in July. Is that right? August, thereabouts.
It's a moving date.
It's a moving date, got it. But she's done a tremendous amount of work in multiple arenas within urologic oncology, and one that I've been particularly interested in is the bladder cancer microbiome. So we appreciate you coming here to share your perspectives.
Thank you. Thank you so much to the panel, and thank you everyone for attending. I hope by the end of this talk, you're as excited about at least microbiome and bladder cancer as I am. And I'll be sharing some of the data that's currently under review, but also some of the data that's fresh from last week. So the slides that says do not copy, please do not copy. No disclosure relevant to the research. However, PI as well as some of the other members do have funding that has supported this research over the last 6 years. So I want to kind of focus on a couple of things, and I'm skipping urine because it's just the amount of information is incredible. But I want to start with the gut and describe what we have found in bladder cancer patients, and then move over to the intratumor heterogeneity in bacteria in bladder cancer, and then really look at the future directions of where we're going and what we're thinking of doing next with Oncobiome. So I don't want to rehash any of the previous gut microbiome and the the immunology and the basic components in cancer immunity, but there are biologically plausible mechanisms that work along microbiome and work along the systemic immune response, particularly along the Th1 immune response, which is very important. I am immunologist by training, so I do have that component of sort of understanding within microbiome that I think is incredibly relevant to bladder cancer. So over the last 6 years, we've been collecting over 152 patients from 2 institutions, fecal samples, and that includes both samples from healthy patients that are age and sex matched, as well as bladder cancer patients that are undergoing cystectomy for muscle-invasive bladder cancer. We also included patients and their partners because we know that the environmental effect of your surroundings has significant impact on your microbiome. So having the ability to test the partner's microbiome and really look at the difference as the patients and the partners transition through therapy is incredibly important. We thank the patients that over the last 6 years have contributed. And we wanted to look at not only to characterize the differences because characterization is important, but also look at the complete responders versus non-responders to chemotherapy. So these patients get chemotherapy within a neoadjuvant chemotherapy before their surgery. And then really look at the changes in microbiome as patients go through chemotherapy right prior to their surgery, because I think that's equally important. Do things fluctuate? Do they change? Are any of those changes significant to predict response? And what we found, and this is sort of what I was talking about, is there is a clustering that's difference between the healthy cohort and the bladder cancer patients, but there is overlap in patients. And I didn't put this on the slide, but a lot of the overlap actually happens in patients who are smokers versus non-smokers. If you truly separate the bladder cancer patients who are smokers versus healthy, there's a clear separation of their community subtypes. And then this can be explained by multiple organisms in terms of the differences in bacterial composition, particularly in our patients with bladder cancer. It was Bacteroides and E. coli that was present in their stool. So then our next question focused, we know that there's compositional differences. What about response status? Does that change the compositional differences? Does it change your response to chemotherapy? And in fact, there is a trend towards difference. For example, if you look at the neoadjuvant chemotherapy patients who had low prevalence of Bacteroides were more likely to respond to chemotherapy. So 12 out of 14 responded to chemotherapy, unlike patients who had high Bacteroides count. It's not an absolute. There's always differences. Patients are not always by the book, but there is a clear trend toward this association, which piqued our interest in terms of what is the mechanism, what is going on. So then we looked at those differences through chemotherapy, right? What happens with those abundances as patients receive chemotherapy? And what we found by the orange lines in this figure is that as the non-responders continue to get chemotherapy, their Bacteroides counts actually continue to go up through chemotherapy, unlike their partners who actually decrease their counts. And people who are complete responders, that Bacteroides count actually drops by about 10 to 20%. What about our mice? What happens to mice? So I work in the lab. I'm at 50% lab-based research. So I wanted to see what happens to mice. And part of our urine model in bladder cancer is to give a carcinogen called BBN. So when we give this BBN to our mice to induce tumors, their Bacteroides counts goes out the window. As soon as we withdraw the carcinogen, the Bacteroides count drops. There's a clear biological mechanism how Bacteroides works. We don't quite know what. And again, this is genus. This is not species level analysis. And there's multiple species within Bacteroides that we have to sort of look into further. All right, then we looked at, can we use this compositional difference to predict patients' clinical response to chemotherapy? And then how does that compare to the current clinical variables that people use to predict response? And this is early data, it needs validation. This is 2 institutions, but you can see it's pretty high. So based on just your stool sample, your poop, you're able to predict your chemotherapy response. If you take your clinical variables, and we know that even tumor molecular factors are not really predictive of response these days. Actually, it's about 52. The AC is 52 for clinical factors. If you take your stool microbiome, you're getting close to 0.88 to 0.9. So very predictive. Don't want to jump to conclusions, needs more validation, but this is a great example of how we can use this to help our patients. Lessons learned over the last 6 years of this collection is that stool collection is preferable over swab samples. Prefer multiple time points if possible. We need more institutions, we need more patients to validate this data to really make it accessible for patients for testing. And then because of the limitations of 16S analysis in terms of species, shotgun approach gives you more ability to look at the species level analysis. Okay, I want to move over to more of the intratumor microbiome, which is what we're focused on now. So many of you have seen studies looking at intratumor bacteria, and a lot of these are done in terms of bulk sequencing. So imagine a smoothie, you throw a bunch of fruits and you get it. And then you're like, oh, great, it tastes like banana and strawberry. But essentially, what is it you're looking at when you're looking at bulk sequencing? You're looking at everything under the sun. And what does it mean in terms of the immune composition? What happens to these bacteria? Is it actually in the tumor cells, or is it in the immune cells surrounding the tumor cells? And just by bulk sequencing, it's very difficult to know what is going on. Presence of bacteria does not imply it's in the tumor. Presence of bacteria within tumor sample implies that it's potentially close, not necessarily within the tumor cells. For example, I want to thank Dr. Alexander Wyatt for giving us this data from the ctDNA positive negative trial. And what you can see is the same patient, 5 different biopsies from the same tumor, had 5 different taxonomic abundances. So just like your tumor molecular heterogeneity, there's bacterial heterogeneity. So by taking one sample and using that as prediction for some sort of response, is that really the right approach these days? And I don't think it is. But you can use single cell sequencing to look at intratumor correlation between the microenvironment and the oncobiome. For example, on the left, you can see the composition of bacteria called Pseudomonas, and on the right, you can see the tumor microenvironment. And what you actually find is that majority of these are actually within the monocytes. Very little, less than 1% of the actual bacteria is actually within the tumor cells. Majority are actually in the immune cells surrounding the tumor cells, and majority of those cells are monocytes, CD14 and CD16 monocytes. So what does that mean to you? Well, we wanted to see what actually happens if you mix the monocytes with the T cells, trying to reproduce that environment along with the JD2, which are your bladder cancer cell line, and see what actually happens. So if you just take your monocytes with the T cells and you mix it in with a tumor lysis, and we specifically chose tumor lysis because it's more, it has more likely caused dendritic cell differentiation. If you just mix your monocytes and T cells, you actually find that majority of the monocytes become dendritic cells, particularly plasmacytoid dendritic cells, which are great type of dendritic cells. However, if you expose those same monocytes and T cells to half an hour of Pseudomonas, so a small amount, subclinical, patient would never even be symptomatic with an infection of Pseudomonas, you almost decrease by half the amount of monocyte differentiated into dendritic cells. And you sort of push that differentiations into macrophage. And even co-stimulation with receptors such as toll-like 2 and 3 receptors really has no ability to modulate that response back to baseline. This is mostly driven, what we found, to be by IL-6 and IL-1 beta mechanism, which makes sense. It's a pro-inflammatory, pro-tumorigenic environment. It causes inflammation, causes macrophage inhibition factor and recruitment, causing more pro-tumorigenic factors. What happens to T cells? Well, you have decreased T cell activation within any small amount of any bacterial, particularly Pseudomonas exposure. So why would we care in terms of us as a cancer biologist? Well, because it's sort of that vicious cycle of you have the cancer cells online with an infection, and even though if the tumor itself is not infected, if you have any presence of, for example, Pseudomonas, you have this pro-tumorigenic environment that unfortunately blockade of IL-1 beta or IL-6 alone is not going to help the situation, because like most tumors, there's parts of it that are incredibly hypoxic. And even if you block IL-6 and IL-1 and you give them therapy, unfortunately, that therapy will not penetrate the hypoxic area. But there are things that can penetrate and actually live and thrive in hypoxic areas, and that's bacteria. So our next goal as future therapy is use bacteria as potential vectors for the hypoxia and introduce bacteria into these tumors where nothing else will really penetrate, no T cells, NK cells, monocytes, and really thrive on the actual bacteria destroying those parts of the tumor. And you can see this is our mouse model, is that by mixing a certain type of bacteria along with certain blockade, you can decrease the tumor burden within 3 weeks of therapy through intravesical instillation. So far, 10 out of 10 mice are alive, unlike the chemotherapy ones and the immunotherapy ones. So I want to thank the team that works with me, including my PI, Dr. Bosch, as well as our funding mechanisms from AUA and BCAN. Thank you.
And we have a couple of minutes for questions here. I see Dr. Thompson coming in the microphone.
I'm just curious. I'd like one of the panelists to teach me what happens when you give IV antibiotics, oral antibiotics, because all of these bladder cancer patients, or a lot of these bladder cancer patients, especially the cystectomies, have all recently received a course of antibiotics. They get their pulse at the time of their TURBT. If they've had 2 TURBTs, they may have had 2 pulses. And a lot of these in the family practice community, they show up with some hematuria and they're treated with antibiotics. They may have multiple courses of antibiotics before they get their referral to the urologist. Will that cause some kind of bias in terms of the interpretation of the microbiome?
So we limited our study to, they're working. I'll just speak loud. So in our patient cohort, anyone who had any antibiotics within 6 weeks of therapy was not able to be included. However, if you look at the studies looking at single dose antibiotics for OR, typically it has minimal effects. However, if you're on prolonged therapy over 3 days, it does take about 6 to 10 weeks for reconstitution. Now, if you're going to reconstitute the same as you have before, likely, yes, if you have not changed your environment. However, sometimes, and I know you're waving no, sometimes you do not. So when we looked at it, and the patients who got unfortunately recruited into the study but had antibiotics, they actually had very similar sort of microbiome, but some studies show that you don't, depending on your environment.
So your multi-institutional trial, the idea is once you do your first cysto and see the tumor, collect the specimen, something like that.
So yeah, and we always collect the specimen before they get any kind of OR antibiotics as well.
Yeah, great, thanks.
Is that antibiotic dependent? So we use a lot of, say, macrodent, macrobid, which is primarily excreted in the urine.
Mm-hmm. So if it's just in the urine, it will not. But if it affects gram-positive, specifically in the gut, it will affect it.
It definitely matters. So even Cipro, if you do a culture in 2 hours of taking Cipro, we're almost culturing nothing out. Whereas maybe nitrofurantoin doesn't have a big effect on certain bacteria. So that's why a lot of these studies are taken when we don't have antibiotics, because it really will change.
But just a quick point.
For the most part, it's cleaner that way when we look at patients that do not receive antibiotics. But that doesn't reflect reality. That's what we've learned, right? And I'm not an oncologist, but I get this question a lot from treating physicians. So we've launched at MD Anderson a randomized trial where melanoma patients that are going to get surgery, half receive ANSEF and the other half does not, to really prevent what I hear from Jen Wargo and many others is a very minimum risk of infection. And we've looked at their stool microbiome, serum metabolites, fecal metabolites. And we do see, depending on the type of lens that you look through, you may see changes in the composition of whatever, microbiota, microbiome, gene expression, secreted products. What we don't know is if that is sufficient to hamper a response to immunotherapy down the line or not. And the other thought is that these antibiotics are not all created equal. As Laura mentioned, you hit Bacteroides, you hit Firmicutes. Bertrand showed us there's some Clostridia that are usually popping up and negatively associated with response. Can we get rid of those? Can we favor the good taxa, the good ecologies? So I think there are still a lot of open questions here. And I don't know if we're going to get to a standardized approach. Patient that has likelihood of receiving immunotherapy, what type of antibiotic you give, I don't think it's going to be as clear as that.
Great, thanks. We'll get one more question. Quick answer.
Just wanted to follow up on the Pseudomonas and the tumor monocytes. Did you test monocytes in other tissues or elsewhere? Is this pervasive? Are these Pseudomonas coming from the gut microbiome and sort of pervading all the monocytes in the body and setting up immune suppression?
So.
And finally, I want to know what coffee does, but.
Coffee does all good things. No one is ever studying that. So Pseudomonas, particularly what we studied was in bladder. So it's likely from the bladder, and these patients had multiple catheterizations. They were asymptomatic. They had no evidence of an infection. However, it does not mean that there's no presence of bacteria if you're asymptomatic with an infection. So it probably got introduced from the environment. I don't think it's a translocation mechanism, even though we do find a pretty good number of Pseudomonas in the stool. I don't think it's translocation. I think it's intravesical at some point during instrumentation or other things that happen.
So thank you. We're going to have lots of time for discussion, I'm hoping, during the clinical trials panel, which is going to happen shortly. I'd like to ask Dr. Wu to actually come join us up here on stage, if that's okay right now. We're going to jump into our last talk, second to last talk from Dr. Liss, who's going to discuss the microbiome and prostate cancer. Mike is associate professor at UT San Antonio in their Department of Urology and is doing a lot of tremendous work in this space.
Thanks. So we'll focus on, I do have a disclosure. I founded a company recently, Oncobiomics, and I'll talk a little bit about what led to that, but not specifically. So when I looked at the microbiome for prostate cancer, it's not as immunogenic as everybody else has been talking about. So this is a harder task. But when I first started looking at this, there was this one experiment by Dr. Erdman at MIT, and she took wild type mice and APC mutant mice and disrupted their microbiome with Helicobacter hepaticus, which is inflammatory. And they found that in the APC deficient mice that when they removed the organs, that they found prostate cancer. They're actually looking for colon and breast cancer. They actually found prostate cancer, which is interesting. And when they treated with TNF alpha, they didn't form the cancers, and the sham mice didn't form the cancers. But what was really interesting was what happened next when they took out the mesenteric lymph nodes and they put them back into either wild type or APC mouse. They both formed prostate cancer, whereas that didn't happen with wild type mice or any of the other factors. So this is quite interesting in that there's something with a predisposed patient that then has a bad microbiome that leads to a cancer, and this is effect of the immune system. And we know this in extremes for prostate cancer, that there's some data coming out with inflammatory bowel disease, and that you have a 4-fold risk of prostate cancer if you're diagnosed with inflammatory bowel disease. So there's definitely some connection here. And that same group, Dr. Kundu at Northwestern, did a mouse model, and they inflamed the colon, and then completely separate is the prostate, which showed induced inflammation with all the players that we think of for prostate cancer development, AKT, NF-kappa B, MYC. They showed more DNA damage in those prostates, and they also showed immune infiltration. So an organ that is completely separate to the prostate is causing these inflammatory effects. And our lab, one of the hypotheses I'm working on would be, okay, how are they connected? And we know that the rectum and the prostate actually share the lymph node chain, and we remove those during surgery. So we took some of those patients that had positive lymph nodes, and we sequenced them for bacteria. And we didn't really show the patients all paired up with themselves. The cancers didn't separate from the non-cancerous nodes. All the patients actually separated together. So we know that the immune system, and we think that antigen presenting cells in the colon will bring these bacteria to the lymph nodes and process them, and then actually train our immune system. So we know that these bacteria are actually in the immune or in the lymph nodes. So it's establishing this new gut prostate axis. We've heard of gut brain axis, and other factors in bladder and kidney. So, what can affect our microbiome? High fat diets, medications. We talked about antibiotics, this dysbiosis factor, and then it actually can produce androgens. And I'll talk a little bit about that later. But most of it is this inflammatory effect that could lead to cancer. So I'm going to talk about 3 distinct areas in prostate cancer where I think the microbiome can have an effect. So one is risk for prostate cancer. So I published this in 2017. We won best paper in European urology, but really it was kind of the first time linking the microbiome to prostate cancer. And so we had not amazing AUC curves, but there was some diagnostic effect, and we could tell the difference. And the top line is PSA density. So we were actually approaching PSA density, which I think is an excellent biomarker. So this was just kind of the first dipping the toe into the microbiome, and does this have some type of relation? And what we did in that study was we actually took their metabolic effects. So you could predict their function in the microbiome. So we took that to see if there was some type of relation to what are the bacteria producing, and is that the cause of these cancers? We showed that folate and biotin, so B vitamins, are being produced by the bacteria, which were great in the non-cancer patients, and that sugars were bad, which is unfortunate for me because I love sugar. But so watch your sugar intake, essentially. But we didn't have great aha moment here. We just knew that there was definitely some effects. And since that time, we've sequenced over 1,400 patients, and now we're building the model. We're getting much better performance to be able to predict out who is going to be forming prostate cancer. So this is immediate prior to biopsy in these 2 groups. But now we have 5-year data out now. It's in my computer that I have to analyze now. So we're pretty excited about that. And then I went back to the inflammatory bowel disease. So that biomarker that we created, I went back and saw, okay, there's some publicly available data from UCSD that on inflammatory bowel disease. And so we used our marker, and we definitely showed that in men, if you had inflammatory bowel disease, that marker was definitely increased. Whereas in women, and we talked that there are some differences, that wasn't the case. So there is unique bacteria, but bacteria can produce different substances. So this was a recent data from the PLCO trial looking at phenacetylglutamine, and that was increased in those who had lethal prostate cancer. And this is uniquely associated with the microbes in the gut. And some of our other speakers talked about butyrate, and we've seen it in cell lines, but we haven't quite gotten to that point to test it in prostate cancer yet. But I think this is an exciting area to think about. So what about androgen deprivation resistance? We all have those patients where we gave and then you get frustrated because their PSA hadn't come all the way down to undetectable, and you check their testosterone and it's still higher, it's 30 or 40, and we're kind of trying to figure out why it's not going all the way down. Well, bacteria can produce testosterone. And so these were a couple of the bacteria that they found could, after castration, go up and then end up making it. And we know tumors can make it too. So they used 3 different cell lines and implanted mice models. 2 were implanted in the tramp mouse, and essentially they showed that if they castrate the animal, then you can get a response. But then if you gave antibiotics, that you got a longer response, or smaller tumor sizes, and the animals lived longer. So in this case, antibiotics were good. You killed the bacteria that were making the testosterone, you got a better response, and the tumors were smaller. And so here they basically showed in patients as well, you can give a tagged pregnenolone, and it can go all the way through the gut, and in the portal vein system, you can see higher levels of DHEA and testosterone. So they actually did isolate that. And then they took humans, and in each of these groups, and I circled the top right corner because that shows that there's hormone sensitive patients, hormone resistant patients, and then patients who died of hormone resistant prostate cancer. So in those patients, you could see higher and higher levels of those specific bacteria that they used in the animal studies. So this would be a great area to study for hormonal resistance. But what about docetaxel resistance? So in this one, they gave the mice, they implanted the tumor, and then they gave an antibiotics, and this tumor grew. Right. So it's not as easy as just giving the antibiotics or checking which one works and which one doesn't. So in this model, at the bottom, you can see all the tumor levels were higher if they gave antibiotics. So now there's something in the system that's actually beneficial. So we have to be careful about giving antibiotics when you're thinking about chemotherapy. So this is extremely complicated, and I think that we're just touching the surface. And so my talk was more on trying to think about applications of how we can use the microbiome in our studies and maybe following them over time. So that was it for what I had. Thank you.
Thank you. And wrapping up our didactics this morning is Mike Wu, who's a terrific biostatistician affiliated with SWOG in our melanoma group. We all know here in the audience with a lot of GU representatives how vital, for instance, Kathy Tangen is to our functions within the GU group. Mike plays that critical role within melanoma. But little did I know he was also an expert in the area of the microbiome. And your name, Mike, has actually come up in multiple conversations that I've had with microbiome experts. So we're really delighted to have you here to discuss biostats in the microbiome.
Just be gentle, please.
Yeah, I checked his slides and he won't roast any of us, but I'm sure they were
not
just
terribly closely. But you know, as a statistician, my default is to say you guys are all wrong. I mean, it's just what we do. All right, so thanks very much for the kind introduction. So my lab, actually 80% of my lab works on the microbiome. I only spend about 10 to 15% of my time doing SWOG, which is more of a hobby than anything else, honestly speaking. But I'm delighted to be here to talk about some of the work that we've been doing. Well, actually, this is not going to be so much work that we've been doing, but some of the lessons that we've learned over the last 10 or 15 years where my lab has been developing statistical and computational tools for analyzing the human microbiome. All right. So thus far, we've been very, very happy so far with all the exciting work on the microbiome going forward. This is my daughter's favorite poop emoji. She calls it Happy Unicorn Rainbow Land. And of course, you can see the fairy buzzing around. There's so much promise for the human microbiome in cancer. Now, welcome to Stats Land, where we tell you what can and oftentimes does go wrong in these studies. And as a statistician, one of my favorite statistics about the microbiome is probably 80 to 90% of what's published out there is not entirely accurate. All right, I'd call it sort of crap, for pun fully intended. So we've seen a lot of associations between cancers and the human microbiome. And the unfortunate fact is that some of these could eventually end up in the toilet. Not all of them. In fact, much of the work is fairly robust here. We are a much more careful community, but there is still a lot of junk done here as well. And there's a lot of problems. So, oh, by the way, don't flush your papers down the toilet. I got complaints from housekeeping last time. So why is it that many of the microbiome studies and much of the microbiome data we have is crap? Well, there's a number of different issues. This ranges from poor study design, poor sample and specimen collection and processing, poor statistical analysis, and ultimately just poor interpretation of results. And what this leads to, of course, is, well, if you have poor study design, then that means you're actually never going to be able to answer the question you're interested in answering. If you have poor processing, then what you're measuring actually is not what you think you're measuring. And this is a real problem in the field with substantial biases. If you have poor statistical analyses, your results are invalid. All right. And if you just don't know what you're talking about, then sorry, there's just no idea. You just got to go home. It's why I'm really happy that we have some microbial ecologists and other folks with us who can really tell us where we're going wrong in these interpretations. All right, so I'm just going to breeze through a few of these. With regard to study design and sample timing, you really have to ask yourself what you're trying to figure out. This includes which microbiome you're interested in, what is the timing of specimen collection, and importantly, what other data types you're going to be collecting at the same time. 10 years ago, if you were studying the microbiome, your RO1, everybody would be happy. These days, when I sit on study section, if you're just doing the microbiome, that's never gonna fly. They're gonna want to understand mechanisms, and you're going to need other things going on at the same time. All right. So which microbiome? If you're dealing with the gut, as we've largely been talking about, that's easy. There's tons of shit out there, and it's really easy. If you're dealing with the vaginal microbiome, also relatively easy. Oral environmental microbiome is a little bit more complicated because you don't know what you're looking at. If you're dealing with, say, a cystic fibrosis lung, again, lots and lots of stuff to swab and to get. All right. Then you have issues of low biomass communities. This includes intratumoral microbiome. All right. I've also analyzed breast milk, the ocular microbiome, which I'm not entirely sure exists, but some collaborators seem to think it does, and the healthy lung as well, which there's not much in there, honestly speaking. All right. And so the challenge here is that when you have very, very low biomass, you're highly susceptible to contamination. So contamination comes from all different sources. And the issue, of course, is that when you have contamination, this is what your true sample is, but you end up with all sorts of other types of minority taxa that are showing up. And so one very sobering story is about 10 years ago, a group at Baylor published this very nice paper on the placental microbiome. And so literally all the OBs and all the folks in maternal child health went crazy. They published tons of papers on the importance of the placental microbiome. A couple years later, all right, what folks figured out is there's actually no placental microbiome in general. All right, and this has been validated numerous times. And the general consensus is that what they were amplifying are just the bacteria that are inside of their reagents. All right, so you have to be very careful when you deal with low biomass out here. All right, it's a real concern. And when you deal with the intratumoral and some of these other things, it's a problem. So be careful. Timing is key. You need to make sure that what you're collecting is what you need. So this is a study looking at GVHD I worked on. And so you can see that the alpha diversity here is dropping in preconditioning, drops to a nadir here, and then sort of bounces back up as you reach neutrophil engraftment over here. And so it's fantastic, but then you need to make sure that you're really capturing what you want to. It is dynamic. And so you need to get the time points. And when you talk about the microbiome at, say, 30 days versus 60 days post-transplant, you're actually talking about different entities in this case. It is highly dynamic. Bear that in mind over here. All right. Yeah, so we'll go there. The other thing is timing relative to other data of interest. This was a poor study design where they collected the microbiota all throughout, but then they only collected hematologic measurements, which they were interested in at certain time points over here, and they didn't line up very properly. And so as a result, some of our interpretations are problematic. So in terms of sample collection and processing, all right, you need to think about everything from your sample collection and preservation kit to what processing pipeline you want to go through. A lot of these choices can have very serious downstream effects, particularly with regard to biases that are induced over here. Another issue that I'll briefly mention is batch effects, which are extensive here. So we've already talked a lot about which kits you want to use. Well, some folks have talked about it, and it just depends on what you're trying to do. If you're doing metagenomics, relatively straightforward. If you want things like metabolomics or metatranscriptomics, metaproteomics, metaepigenomics, you need to be a little bit more cautious as to what you're actually looking for here, right? And then one of the things that we found, and this is something that has been very sobering to me over the last few years, is that biases occur at all steps in the process of your data collection here. All right, so here's an example where you might have a mock community where you know the composition is 33%, so a third of each of these different bacterial taxa. All right, the fact is that you accrue biases as you do extraction. All right, you got to think some cell walls are easier to break down than others, but if you use with some kits, but then those kits may also end up destroying some of the DNA. So there's all sorts of things that happen there. You have PCR amplification bias, sequencing bias, as well as bias in the informatics protocols over here, such that what you end up with is a poor representation of what's truly there. All right, so it's a real concern, and this is something that is observed there. The problem is, so here's just another example. These are true mock communities, all right, where this is what the diagonal line is where all of these dots are supposed to be, and you can see they're totally different. Each of the different colors represent different bacterial taxa. And if you look over here, the bias differs based on different bacterial taxa over here. And so it's taxon specific, it is unpredictable in many cases. And so what you're measuring oftentimes is not actually what you think you're measuring. And this is a real problem as we try to understand exactly what is happening in these communities here. Right? So be aware of that, and just remember, it's a caveat to all of the studies that we are conducting here. Batch effects are real. There's some of my collaborators think it doesn't exist. I showed them differently. This is a study of cardia where we had 650 participants sequenced across 6 different batches. And you can see that there's tremendous separability here, both before and after normalization for sequence depth. And that persists throughout. It is a real issue. If you actually have a huge sample, if you actually have a very small sample size, you sometimes don't see the batch effect, but it is still there. So just be aware that it exists and keep that in mind. I'll briefly talk about statistics here. All I'm going to say here is really that virtually most of the methods that you see tailored to microbiome data, they're all invalid, actually, to the point where they can give 80% to sometimes 200% false positives. It's a really big problem in this situation, but yet folks seem to like these approaches. So with regard to, you get significant p-values, and so you go home very happy, but the reality is a lot of what you're finding is junk. All right. So what works? Actually, it's kind of funny. The really, really old statistical approaches developed in the 1920s actually end up working. The approaches that don't work, the fancy approaches that we've been developing over the last 10, 15 years for, say, RNA-seq analysis. One major take home, if you get nothing else here, don't use DE-seq2 for your microbiome data analyses. Your false positive rates can be 200 to 300% in those situations. There's also these really weird methods that one of my good friends, Curtis Huttenhauer, has developed. He named them all after bread. And I'm just going to say that all of those approaches, some of them are actually valid, some of them are not. It depends on how you're using them. And it's also problematic here. So I'm basically at time. So I'm just going to end by talking a little bit about actually, I'm going to show this first. Other people have recognized the invalidity of most of these methods. I'm not alone here, and more papers are coming out all the time. But nobody likes listening to us statisticians because, again, we're just bringing bad news. All right, in terms of interpreting results, one thing to remember, microbiome, everybody seems to treat it as either a cause or an effect. The reality is we don't know. And there are some bugs that may be causing, some bugs that may be resulting. And again, it's very difficult to tease apart. Sometimes you're just asking too much from your data when you want to try to do that. We don't have enough data to say in many situations. You have to really run experiments to tease that apart separately. Microbiome is heavily confounded. We know it's related to age, sex, gender, anything that you want over there. All right. It's related to smoking. It's related to diet. It's related to your ancestral background, everything. And so there's tremendous degrees of confounding. And I think that much of what we see out there is due to substantial confounding. And ultimately, you need to understand the mechanisms. This is extremely important as we start doing bugs as drugs. All right. FMTs, I think, are an extraordinarily crude tool. I've run studies where they are positive. I've run studies where they are negative. And the fact of the matter is it's very difficult to figure out why. And until you know what's going into there, all right, it's never going to be something that can be really mass replicated, I think, outside of certain specific conditions like C. diff. So things I didn't talk about here, the metagenomics, transcriptomics, epigenomics all bring unique challenges. Data integration is where most of my lab works. And also remember the microbiome includes other strange and wonderful things like viruses, archaea, and fungi. And so take homes here, microbiome's exciting, but the data are problematic in many situations. What's the solution to this? Come talk to your statistician. If you do it too late, then I can give you an autopsy on your study, but that's about it. All right. So come talk with me. We have solutions to some of these. I'll stop here. Thank you. And I'm sorry for going a little bit over.
Perfect level trade places over here.
Now you know why we put him last, so we could keep everybody engaged and then just crush us down.
And Kathy, you would tell us if you thought our studies were crap, right? You would let us know. So we have a couple of minutes here for Q&A. I saw a couple of people getting up to the microphone earlier, so please feel free to do so. Why don't we start with, can we start with Nicole, perhaps? Dr. Kuder? Yeah, go for it. Go ahead.
Thank you so much. Hello, Nicole Kuder, Seattle. A phenomenal panel. Thank you so much to everyone. Please do not get discouraged. These are thank you, Mike, for your frankness as well. We're so grateful to have you. And this, I can say the same was in the early days for the genomics era, and the genomics era is now revolutionizing oncology. So thanks to methodologists like Mike, we will get there. Mike, can I ask you, how have we standardized processing at SWOG, and what should be a SWOG investigators consider? Or do we need to do more standardization even within SWOG, despite having you and other wonderful experts involved?
With regard to the collection and processing, I don't know that we have exactly standardized protocols. The issue is within SWOG is that this is typically done at individual sites or by individual PIs and investigators there. And so we're sort of beholden to their preferences, whether they want to do 16S or metagenomic sequencing. I have my preferences, but cost comes into play as well, and as well as just sheer feasibility. So it's whatever the investigators' cores are doing.
So whatever you can do to help us, to be really fully on target, we really appreciate it.
You throw me some Benjamins and I can happily do things like that.
Dr. Tripathi.
Thank you so much. Absolutely fantastic presentation. Quick question for Dr. Buchanan, I think from your field standpoint. So the tumor microbiome is quite interesting because we know very little about. Do you think in your clinical cohort of patients, did you see any differences with regards to clinical characteristics like upper tract or lower tract, concerning the urine dynamics may be contributing to the tumor microbiome? Interested to hear your thoughts.
I did not look at upper tract. In terms of urine, going back to our statistical talk, and contamination is that if you get voided urine, you get significant amount of contaminants. So we actually stopped after about 10 patients and we started getting only catheterized urine. So that's a huge confounding factor that I would urge people going forward and collecting urine just to get catheterized samples if possible, not voided. For urine specifically, I didn't present data, but lactobacillus was one of the sort of kind of like a Bacteroides was for non-responders. Lactobacillus was present more in responders, and it was a marker of minimal residual disease in those patients.
Great, thank you so much.
Quick follow-up, maybe a
Parallel with stool. Stool is really not representative of anything in the GI tract. But we use it as a proxy because it's cheap to get. It's not invasive, et cetera. And when I started in this field, I ran a study with a collaborator where we looked at the differences in the GI tract from proximal to distal, all the way to the fecal samples, and we saw topographic differences, compositional differences. And just this week, there were 2 papers published back to back with a beautiful commentary. I think it's titled "A Trip Through the Gut," where 2 different groups gave patients, or a big group of people, smart pills that could sample across the GI tract. And they ran metagenomics and metabolomics. And you could see by compartment, very distinct signatures. So that's another big confounder in our field, that we're using stools as a proxy to the entire what's happening in the entire GI tract. And I'm guilty of this. We build networks. We think about ecologies, statistically driven ecologies, right? These are not, we're not seeing these bugs based on what we see in stool. And that is likely not representative of what's going on in each compartment of the GI tract. But we do what we can, right? And we start here and then we move up in a sense.
I think we have time for 2 questions, if we have brief responses to those, if that's okay. I'll start with Dr. Thompson.
I'll try to be as brief as possible. Mike, your talk reminded me of something we did back in the EDRN a number of years ago with Zeding. And perhaps 15 years ago, there was this blurb of interest in something called CellDTOF. You know where I'm going with this, right?
Yeah.
And so the challenge with it is proteins they disappear at different rates based on processing. And there were these remarkable data on the association of CellD and cancer. I mean, ROC curves were off, and we put together 3 separate laboratories with CellD machines cranking out with the same specimens that went to them, cranking out the same results, and we were completely unable to determine that the biomarker was effective. However, which something that's not in the paper, Zeding told me that the one thing he could tell was which institution the specimens came from. And so I would imagine it's just like protein decomposition, the moment that you draw it or whatever, I suspect that these biospecimens will change. Do they change? And if so, that adds another element of complexity in specimen processing to be to be able to replicate it from institution to institution, multi-institutional trials like SWOG?
Yeah, we did some studies on, actually, you can collect it from your digital rectal exam if you use 2 gloves. We did glove tip, we did swab, stool. We actually could get similar results, but it depended on, well, when did you get it? Did the patient get it at home, and did it go in the fridge, or how did you collect it? Which collection kit did you use? So it's like almost an infinite amount of variables that go into that collection. So as we design them for SWOG studies, we really have to talk about that before we do it.
Dr. Petrolak.
So 2 comments. Morning. Great presentations. Really enjoyed all of them. We often run into the clinical situation where a patient has a non-castrate testosterone level, and you've given them an LHRH, you switch them from an antagonist to an agonist, or back and forth. From your presentation, what you mentioned about the gut biome, is this really significant enough to cause a elevated testosterone level in the serum?
So they did patient studies, and it's going to be about 20. And that was exactly my question to them too. It's small amounts, but in some patients it was actually quite large amount of testosterone, depending on how much bugs they have. If you have it present and there's not a lot of it, so abundance matters there. But that's still an early. The fact that it's making it, and that could be a cause of some of these things, was a little shocking. So,
and the last comment, a little bit food for thought. For years, we've been told that when you culture a conduit, or you culture somebody after a cystectomy with their diversion, that if it's colonized, doesn't mean anything. I think we have to really start rethinking about that now.
Correct.
And in terms of adjuvant therapy for urothelial carcinoma using nivolumab, the question is there any difference in the overall outcome based upon what type of diversion these patients have? And I think that's going to be a great question for thought later.
Thanks.
Great point. Great point. I'd like to give a round of applause to Dr. Rudy, Dr. Bukovina, Dr. Ajami, and Dr. Disman for great presentations. And we'll excuse you guys. I'm actually going to see if we can keep Dr. Wu up here on the stage. So if you guys want to have a seat, I'd like to invite up Dr. Kuderer, Dr. Reckkamp, Dr. Lee, and Dr. Barada for our microbiome panel discussion. So the format here is very different. We've actually given each investigator about 5 minutes to discuss their particular interests, perhaps as they relate to the microbiome, perhaps not. You'll see, for instance, Dr. Reckkamp is a champion for a completely novel clinical trial design called Pragmatica that I think may bear some relevance to what we're doing within the GU group here at SWOG. But ultimately, these 4 presentations are actually meant to really foster a lot of discussion around clinical applications of the microbiome. So let's go ahead and queue up the slides here. I think we're starting with, do you remember, Mike, whose presentation is first here? Maybe I'll let you access it.
Well, it's such a pleasure to be here, and thank you for the invite. And it's my pleasure to represent the whole large team of S2013, which is an immune checkpoint inhibitor, a large prospective observational cohort study. And as we only have 5 minutes, I would like to very briefly already upfront summarize what this study is all about. It really is about to develop clinical molecular predictors for immune-related adverse events. That's our overall goal. The study is for patients that are receiving the standard of care for immune checkpoint inhibitors in solid tumors. So it's a real-world population that's readily available to all of us in clinic. So this is combining a real-world toxicity cohort with then complex translational data. We're not only collecting longitudinal IRAE data, immune-related adverse events, but also associated longitudinal patient-reported outcomes, as well as longitudinal blood. And now we're excited. We just got IRB approval to add the microbiome. So this is a very translational, but otherwise a very parsimonious real-world cohort. And we're very excited. We were not sure how feasible this actually is. So we started first with blood, then with tumor, and now adding the microbiome. And we're very excited to report very strong accrual. And we think we have now also, it most likely will expand to include chemo checkpoint inhibitor combinations in an expansion cohort. So very briefly, our primary goal is to develop and independently validate a risk prediction model for grade 3 or higher immune-related adverse events in the solid tumor setting. Again, not to go into details, we have some key other secondary objectives, which we will not discuss, and translational objectives, which are, first of all, we want to assess and validate the cytox score, which has been implicated to be a predictor for immune-related adverse events, which is a very simple a cytokine score, much simpler than these complex omics discussions that we're having as well today. And also to establish a repository to really be ready for the future, to have tumor and blood and plasma, including Buffy codes, so also for germline, as well as now adding the microbiome to assess what are these normal biomarkers potentially telling us. So the schema, it's patient gets assessed at baseline, they start therapy. So at baseline, they get these tissues were collected, then they start their therapy, and then especially at 1 month. So I want to highlight that 1 month time point where we collect things again, especially blood and microbiome and PRO, to really assess these early changes that also were hinted at by other colleagues that may give us really strong hints about the future development of toxicities. So these are over 2,000 patients that we're accruing. Early on, we were in the 40 range. In the second year, we ended up in the 50 to 60 patients per month. And now we're excited, we're up to 80 to 90 patients per month accruing. So thank you very much. Please reach out to us. We are happy to answer any questions, and I look forward to discussing also how we can operationalize this best for patients.
Dr. Barado. Dr. Barado will discuss the RCC trial.
So this is interesting. We're going to sort of pressure test this trial design with you guys. This is something that our renal cell subcommittee came up with, led by Pedro and Alka, in the microbiome space. So it's a trial of Cabo and Nivo with this probiotic that a live bacterial product that Nasli had referred to earlier today. Pedro?
All right. Thank you so much, Monty. Good morning, everybody. What an outstanding workshop. I'm sure Nick would be very proud. And thank you for the opportunity to be here. So I'm Pedro Barata, oncologist, University Hospital Cytometry Cancer Center in Cleveland. And I'm just very happy to be involved in this project. I mean, we heard a little bit or quite a bit about CBM-588, great work from Navli and Monty and the group. So we know immunotherapy-based combos are standard of care for patients with metastatic renal cell carcinoma. And we also have seen all morning data showing as an association of microbiome with outcomes for patients on checkpoints. Specific about CBM-588, we heard is a live bacterial product. Includes Clostridium botulinum, and that can augment the activity of checkpoint inhibition. When we do have data, we had data when we put this together in small cell lung, and what I'm going to show you in the next slide is actually, what I think we've seen earlier today as well, but also data in renal cell carcinoma. So really wonderful proof of concept trial from Monty, Dr. Disman and the team at City of Hope. We actually ask a very pragmatic approach of what, what patients would do, adding to a standard of care at the time of Ipi/Nivo, combined with CBM-588, as far as not only, as far as clinical outcomes, but also very interesting, correlatives in here. And as we saw earlier today, the data is very compelling, favoring the combination of CBM-588 with Ipi/Nivo, right? So this data has been presented and published. Again, great work. And what's impressive is, they didn't stop there, right? So the landscape, it continues to change. Now there's a standard of care for frontline. We go beyond Ipi/Nivo. We do have another of IO-based approaches that include a TKI with a checkpoint inhibition. So we have Cabo/Nivo, oxipembrol, and FPAM. And so, one of these IO TKIs, Cabo/Nivo, is one of the preferred frontline regimens. In general, these IO TKIs have a higher response rate, kind of similar CRs, but much higher, probably ballpark around 20% or so more response rate compared with Ipi/Nivo, and definitely interested in a preferred combo for patients that come to us with advanced, particularly clear cell RCC. And so with that said, Monty and the group put together another wonderful proof of concept study that I'm not showing you now because it's embargo. The data is going to be presented in 2 weeks in ASCO, and we've seen the data, and basically the data, I think I can say that is pretty much in line with what we've seen with the study we just saw with Ipi/Nivo and CBM-588. So it again explores the same CBM-588 combined with Cabo/Nivo, and the clinical outcomes that were reported. Patients continue to do very well, particularly with that combination. So we do have now data in the renal cell carcinoma arena
arena, not just with Ipi/Nivo, but also with an IOTKI, in this case, Cabo/Nivo, in this proof of concept study that I just mentioned. So we kind of wanted to take the next step, and we thought of, of a larger study to prove this concept that in addition that modifying the microbiome can actually improve outcomes. So we put together this phase 3 trial design in patients with clear cell renal cell carcinoma and previously untreated for their disease to be randomized in 1-to-1 fashion to either Cabo/Nivo. This is a standard dose of 40 milligrams daily with Nivo, flat dose every 4 weeks, with or without CBM 588, which is what has been investigated in Monty's trials, if you will. And we have progression-free survival as a primary endpoint, and we are elaborating a little bit on what correlative endpoints we can include and questions we can ask, keeping in mind all the feedback we got this morning. And with that said, just again, a privilege to be involved in this. Congrats, Monty, Ulka, and SWOG for pioneering really the development of important studies in this context for our patients. Thank you.
Thanks, Pedro. Yeah, the timing of this worked out great because we'd just been through the RFP process. We're going to do questions right after the end of these two other brief presentations. Next up, we have Rose Lee, who's a terrific junior investigator at City of Hope. She's a real dynamo. She actually finished her residency at UCSF and came and joined us. And I looked over her publication list. Tanya, I'm sure you felt the same way. And she could probably be a full professor. Well, it's pretty remarkable what she's done. And she's actually working on some cool concepts in the radiotherapy space across multiple tumor types. Rose?
Thank you so much, Dr. Powell and Dr. Liz, for having me. And just in the spirit of this Dr. Vogelzang Symposium, I didn't have the pleasure of meeting him, but I did have the pleasure of meeting Dr. Powell. He was an amazing mentor, and I think really follows in those footsteps. I'm really grateful and to be here. So I think I'm the first person to mention the word radiation today, not so surprisingly. So as most of you know, radiation is highly cytotoxic. And so in a similar space to systemic therapy, we think about it having an effect actually quite largely when we talk about the pelvis and radiation to the pelvis. And so one of the first things I thought about when I think about the microbiome is how does radiation affect the gut microbiota? And we, of course, as a part of treating a lot of GU malignancies, rather render the mass majority of the gut dysfunctional during the time of radiation, but actually for a significant amount of time thereafter. And I've actually shown a little diagram here on the right, some data collected within our own institution in collaboration with one of our GI radiation oncologists, looking at toxicity chemoradiation and different time points and species diversity, trying to do early stage gut microbiome analysis, looking at correlation between baseline, post-chemo, and then different time points after chemoradiation and different diversity of the different species. Trying to demonstrate that here you can see that this top species is showing that maybe in patients with no grade 2 toxicity, there seems to be higher baseline levels of Ruminococciciae, and then the patients with higher toxicities, maybe there's a lower level of this bacteria, and then the opposite for the species at the bottom in terms of response, and that poor response is correlated with increased abundance of this particular species, whereas good response is decreased. Now, these are very early stage data, but we were really intrigued by these findings and think that they may be generalizable, maybe across even different types of cancers, thinking that maybe radiation has a broader effect. But the data for radiation is really limited in GU malignancies. There is some early data now for colorectal cancer. So we're really excited to try to actually do these exploratory studies. And so there are several studies ongoing at City of Hope looking at the effect, both in these colorectal cancer patients, but also in prostate cancer with Dr. Dorf's mentorship in patients who are receiving hormonal therapy and radiation with prostate cancer. And so today I'm actually going to talk about this hypothesis that I had when I started at City of Hope, which is that, we talked a little bit about diet today, how a time-restricted diet, which is also known popularly as intermittent fasting, may actually modulate radiation response. This hypothesis being that fasting can actually provide a period of quiescence for the normal gut tissues, and therefore allow for improved repair in normal tissues, but actually increasing susceptibility of tumor cells to radiation. And so, without spending too much time on this, we designed this study to randomize patients who are getting pelvic radiation for different malignancies, including prostate cancer, but also gynecologic cancers and colorectal cancer who are getting these large-scale pelvic radiotherapy to either nutritional counseling or intermittent fasting. And so this is a small single institution, but randomized study. And then what we're really trying to understand is whether this intervention is going to have any benefit in terms of reducing toxicity, but also potentially increasing therapeutic response. And so this is kind of the study timeline. And as a part of the study, we are collecting the stool microbiome at different time points. You can see marked in this red little beaker, both at baseline, before intervention, then starting at intervention, before radiation, midpoint at endpoint of pelvic radiation, and at 3-month follow-up. And so one of the reasons we decided to look at the microbiome is that it's a chicken or egg question, right? Some people always think that it's an intrinsic effect, that radiation's toxicity may be actually directly to normal cells, which was my original hypothesis. But an alternative hypothesis is that maybe it's not intrinsic to the human being at all, that it's actually the microbes that are being damaged. And it's very reasonable to think that because we have very heterogeneous response in different bacterial species, and we're intrigued by the preliminary data we've seen in the colorectal patients. And so with that, I just want to thank everyone, especially my group, my lab, who's been doing some of the analysis and collecting some of the samples. Dr. Powell for supporting. Dan Mercier has been doing a lot of the sample collection, and happy to take questions or suggestions later.
Great. Thanks, Rose.
We just have one more.
So it's really a privilege to introduce Dr. Karen Reckamp. So Karen was actually in the office right next to mine at City of Hope for many years. So I always like to think that I learned a ton through osmosis. And she really has always been a terrific mentor and guided me through many difficult career situations. And I've invited her here today to talk about Pragmatica Lung, which I think is something that we're going to see a lot more of within SWOG. I mean, Lucky, maybe later you can comment on this, but it's just a design that really appeals to those of us who have years have been saying, we've got to sort of shed all the sort of unnecessary elements of data collection and studies. And I thought that it doesn't necessarily tie directly into microbiome, but these principles are ones we can probably employ in many of our microbiome studies moving forward. Karen.
So yes, thank you for inviting me to speak today. And when Monty called and said, can we do a pragmatic trial in studying the microbiome? I thought that's an interesting question. And I don't know if you can collect stool and be pragmatic at the same time, but but again, it is, as everybody has spoken about today, it's the simplest way to kind of understand what is going on in the gut there. And some of the things we've heard about today, Dr. Kurdero's study, these are kind of real world things trying to get at looking at toxicities, looking at efficacy. And so I think there may be ways to think about it. And this is a design for a very specific study, but you have to think that it's really, as Dr. Paul mentioned, it's about cutting away what's not necessary and how can we study to get a very specific endpoint. So this is the S2302 Pragmatica Lung study. And my co-chair is Dr. Dragnev, and Mary Redmond is our statistical chair. We have a lot of people involved in making sure that this is going to be successful. So the background is probably one of the most important things about this study. So first off, we're doing this in a setting where there are very limited options. So this is non-small cell lung cancer following immunotherapy or combination or sequential. And though there's great benefit from immunotherapy up front, most patients have progression. And following progression, unfortunately, we're still dealing with docetaxel ramucirumab as the standard of care FDA-approved regimens. And so we have a lot, there's a lot of room in that area to get better, less toxic and more efficacy in the options. So then we did a trial through the LungMap platform and presented this at ASCO last year, which was S1800A. This was a randomized phase 2 trial of ramucirumab and pembrolizumab versus the standard of care. And the standard of care included single agent chemotherapies, including docetaxel and docetaxel ramucirumab. The majority of patients did receive docetaxel ramucirumab in that, and there was an improvement in overall survival with a hazard ratio of 0.69, and the improvement was 14.5 months versus 11.6 months. So pretty robust difference in those groups, but it's a phase 2 trial. And so it hasn't gotten necessarily the traction to move forward as the standard of care. And so it was kind of agreed upon that we wanted to see a bigger trial, a randomized phase 3 trial. But how to do that? Okay, these drugs, we have 2 different drug companies. We have drugs we know a lot about. And I think that became the most important thing. So we want to look very specifically at overall survival. And we have drugs that we know a lot about, treated many patients across multiple tumor types with multiple FDA approvals. So we know about the toxicity. So this was really designed to answer one question, and that is overall survival, and then to reduce the burden of clinical trial participation for patients and for staff, and to increase inclusion. And so this is the simple design. And again, it's randomizing patients to standard of care versus ramucirumab and pembrolizumab. Standard of care is determined by the treating investigator. So we don't prescribe a list of standard of care regimens, but we do guide them to the NCCN guidelines. Part of this was because in the discussions, the thought is there are multiple phase 3 trials in this area that may read out and the standard of care may change. And so we didn't want to prescribe that to anything specific. The ramucirumab and pembrolizumab are provided for those in the experimental arm. And the primary objective is really to look at overall survival. And then our secondary objective is to summarize serious and unexpected high grade, greater than grade 3 treatment related adverse events. So just a small fraction of those adverse events that you would normally capture. And this is kind of a boring slide with the study calendar, but the purpose of this is that this is the whole study calendar. So patients go on to the study as they would for basically standard of care. So we don't require labs for screening. We don't have rhesus reads. We don't have scans. They're done per standard of care because we aren't looking at progression. So the other piece of the phase 2 trial is that progression-free survival and response rate were not different in the control arm versus the experimental arm. So we're really just trying to get to survival. This is just a listing. These are all the case report forms. There are no more. And so the focus is on, we do collect information at eligibility about prior therapies, PD-L1 status, genomic alterations, and we ask to upload a pathology report. But other than that, and then there's the treatment summary forms and the vital status forms that we're collecting, survival, and then the adverse event forms, which again are just high grade. And so again, the purpose is to get to the bare minimum. And this was a collaboration with CTEP, the FDA, trying to make this happen. Some of the early discussions were, if it's not making you uncomfortable, you're not doing it right. And so this, I mean, even when I talk about this, it's uncomfortable. What if we're not collecting the right things? You know, I mean, all the things we've been doing for, I've been doing this for 20 years, and this is very different than how we normally do things. And so we're learning as the study opened in March. We have more than 15 accruals already. And over, I think it was like 180 sites are open. Most of them are NCORP sites. And over about 375 are pending opening. And so this is where sites want to go, patients want this. So how do we figure this out? And even if we have to collect stool, can we still make it more pragmatic? And I do think your study with the CBM and the Cabo-Nivo does fit kind of this. These are drugs we know a lot about. Could we do this more pragmatic? But you do have to take away something. So we talked about PROs in the study. We took those out because we were trying to limit what patients were needing to do. And so you do have to give and take in some of these. They're not going to be the most translational studies, most likely. So I think that's it.
Great, thank you. So now we'll open it up to questions. I did want to shout out to S1600 too, if Jill and Janet are here. They're doing some microbiome work in the immunonutrition and bladder cancer. So hopefully we'll get some results from that too. So,
Bertrand.
So I'm very excited to see that a lot of oncologists now are eager to work on the microbiome. So first, for the CBM trial, I was talking with Monty. So what about the dosing of the bacteria? Because the first trial were done due to or FDA-approved dosing, but if you increase the dose. Second point is that I want unfortunately to remind that the primary endpoint of the first study was not reached, the bifidobacterium. And so I urge you guys to find a biological surrogate marker of the efficacy, and by maybe pooling the two data, even if our bioinformatician would tell us that pooling is not good. But nevertheless, we have to find things that maybe by pooling the two data together, if you have the metagenomic data, then maybe you can pinpoint, because I think we need surrogate marker, and to embark in the study of 842 patients, it's extraordinary, but it would be nice to have upfront maybe a surrogate marker of a gut microbiome endpoint. That would be secondary, but it would be nice.
Yeah, no, those are. Yeah, no, those are fantastic points. I guess we took a pragmatic approach, right? We saw that. I think this is the most developed one on the clinical setting with this presentation, CBM-588, and also with that dosing. So we don't want to invent the wheel. That's, we now have two studies over 50, 60 patients where that benefit remains there. So I'll let Monty probably comment on what his observations were. And he started enrolling patients on that with that dose by twice a day. Regarding the endpoint, also is a clinical endpoint, right? To your point, exactly right. And that's, although it was not the primary endpoint on the earlier studies, if you will, both Ipi-Nivo and also Cabo-Nivo, the clinical activity is there, right? Patients definitely are doing better, not only PFS but also response rate. But I would argue in our RCC in general, for a larger study, we do care about time to progression more than perhaps about what's going on in response rate. But I think your point is really, really well taken. You're right. I don't think in Cabo-Nivo, I don't think I can disclose that. I'll let Monty tell us what we can and cannot say for what he's going to present it in 2 weeks. But you're right. At least with Ipi-Nivo, we did not see an association with Bifidobacterium, for example. So I couldn't agree with you more. I think we are going ahead with a pragmatic approach. In general, the SWOG community and the NCI community has been able to enroll quite well in the frontline studies. I think Pedigree is just an example of that, especially the IO-based approaches where they have around, close to 30 to 40 patients enrolled a month. And, and so that would be my comments. But with that said, I couldn't agree that what you're raising as important points. I think we're going back and we're going to brainstorm a lot how we can incorporate some of those correlatives into the design for sure. I don't know, Monty, if you agree.
I'll make just a quick note on dosing. We did just receive funding recently to explore higher doses of CBM-588 as we're doing a small dose escalation study. I'm a little conflicted. If we run with the dose that we have so far, we've demonstrated efficacy across 2 studies now, right? So I think it's reasonable to stick with the dosing we used originally. I don't know that there's necessarily going to be a dose response with CBM-588 in the small escalation study. I doubt we'll be able to demonstrate that, frankly. But we'll see. We're going to have to make a decision around dosing fairly soon, though, I think. And I did want to give Mike a moment to comment on the idea of maybe combining data for 2 separate studies.
I think combining data across studies, it's something that we've done. The fact is, though, that the variability between study tends to vastly dominate any biological signal of interest. One group I work with is the HIV Real Analysis Consortium, where we're looking at HIV patients. And honestly, there's huge signal. But the variability explained by study to study variability is about 40% of the variability in the data. If you compare that to the variability explained by the variable interest, it's usually 0.5% of the variability in the data. So it's vastly dominated. I actually just did want to turn it around, though, and say, what sort of surrogate endpoints were you thinking of in terms of surrogacy here?
So first of all, it's a microbiome-centered intervention. And in the first Nature Medicine paper, we don't see the change in microbiome over time. So is it the bacteria, does it change the microbiome of the patients? And that's the easy question. And then the immune surrogate marker that's deep down the road, the metabolomic pathway. And because, this bacteria, does it work for every patient, or some patient, do they need it, or they would respond to Cabo-Nivo upfront? And I think I understand, of course, we're here to cure patients, and that's, of course, the primary objective, but we also have to think what we're doing, what's the mechanism, and in these wonderful trials, take time to, pinpoint the mechanism, which patient benefit, could the bacteria worsen the outcome of one patient? We don't know on 20, 40 patients, we don't have the signal, but can we be certain of that? And even if you have a lot of ideas about not combining, I think it would be very interesting to at least do metagenomic using the same metagenomic sequencing. You collected the sample, hopefully in the same way and in the same institution, and then you can combine or look at the difference based on metagenomic data available online. Again, I think that metagenomic is different than 16S. It's okay to combine metagenomic data. It's okay, it's not ideal, but again, we don't live in an ideal world. And we have to be also very cautious to not be too perfect, because if we're too perfect, we don't do anything. So I think that with all the limitations, and before you punch back, with acknowledging all the limitation, I think that they have a lot of wonderful materials that need to be more analyzed and compared to other data online.
So I'm just going to begin by saying Vince Lombardi once said, perfection is, of course, impossible, but we should strive for it. And in doing so, we might achieve excellence. And that is what I continue to believe. But that being said, I'm not opposed to combining studies necessarily as secondary analyses, but I just really wanted to clarify what you're talking about here is not a true surrogate endpoint in this case. You're really just talking about secondary endpoints that we can assess. And that I think is totally reasonable. I mean, when you're talking about surrogacy, oftentimes we're talking about an endpoint that stands in place of something with clear clinical benefit. And so, ultimately, this is a clinical trial where we care about clinical benefit. And so I think that's just an important thing to bear in mind. So secondary endpoints, sure, I'm all for them to try and understand what actually happened when we see or don't see efficacy. So, yeah.
Maybe along those lines, to encourage you for a check, it'd be particularly, again, as a baseline in 1 month, time points, both blood and microbiome. And I was very excited to hear about your data that you think there's really an opportunity to already maybe approach a correlative, a blood correlative for the microbiome. If we could, use iCheckIt on those studies to really drill into that, I think that would be very interesting. And maybe at some point we'd say we have a reasonable blood immune activation correlative. It'd be phenomenal. Early days, though.
I wonder, Nicole, could you and Mike maybe share some of the methodology for the microbiome-based studies you guys are doing? Where is the data being analyzed? What are some of the methods you're employing?
I have to be honest, I actually can't remember. I work on enough studies that at some point I lose track of who's actually doing the processing in this case.
Okay.
In terms of the methods, we're using a combination of what is standard, sort of default standards in the field, for better or for worse, as well as some things that we've developed in-house. I mean, one of the things that we found is that, there are very few statistical approaches for analyzing the microbiome with regard to survival endpoints and things like that. With support from the Hope Foundation, we've been developing the tools for doing such things. So
I do think it'll be nice to kind of harmonize those maybe across our studies as we move forward, right? Yeah, Dave?
Yeah, I wanted to echo the praise for this session, and also Monty for your clinical trial in Nature Medicine. I thought that was a beautiful study, really got me excited. I shared it with everybody I knew. And I think this study is really cool too. It looks pretty easy to do. It's big. The question is there a best practice you can use for sample collection that we can all agree on that can be integrated into a study like this? Because the emphasis really has to be, I guess, on uniform and easy sample collection. What would be the site that you'd want to sample? Is tumor a reasonable surrogate? Your urine is challenging. We're not going to be catheterizing people, I think, routinely in a big clinical trial. Can you just comment, maybe the experts in the room, about what you think the best standard sample collection approach would look like in a study like that?
Mike.
And then you can have the analysis done at,
I
one
mean,
place, but
Yeah, the interesting thing is different groups have shown different results. There's some groups that show it actually doesn't make much difference how you're actually getting the stool specimens. And then there's others that seem to show it makes a tremendous difference. And so it's a little bit difficult for me to say specifically. I know Rashmi, what's her name? I forgot. Has done quite a bit of work on that at NCI, and they've tried to standardize some of the protocols within their epidemiologic cohorts. And I think that's where some of the best work is. But I think it's been sort of mixed, has been my understanding. You may know better.
I think, Michael, you're referring to Rashmi.
Yeah.
Rashmi's work. Yeah. So in the microbiome field, early in the days, HMP 1 and 2, Dirk Givers and Rapnik, Xavier over the Broad, and many others launched this program called MBQL, which was how do we standardize microbiome sampling across the world. I'll tell you, I've in my different positions in this field, probably collected over 100,000 different stool samples and did as much as we could with them. 10 years later, what I do now, I collect stool samples fresh. So I have a specific team that will set up workflows to collect samples from inpatients or outpatients, fresh, ship them over, ice blocks. We do a lot of stringent QC to make sure that those samples arrive at the right conditions. And then we make 10, 12, 20 aliquots. We do RNA later, we do fresh frozen, we do anaerobic PBS, we do a lot of different things that will enable different downstream applications. It's not the same aliquot that is going to serve all the purposes, right? Unfortunately, but there's not a simple answer. There's not a simple method that works for everything either.
Bertrand, maybe you can comment on this too, because we actually have partnered up recently on the study looking at Camu, as Bertrand had alluded to, and we got their protocol from Canada, and it alludes to maybe a more, if you don't mind me calling this labor-intensive stool collection process versus what we're used to using.
So what is described, it's ideal. And this is what also we do in Montreal, but I think to make it possible for every center, it's easier to have pre-labeled tube with RNA later or whatever company you want to work with, or even prepare your own tube and then provide this. But I think it would be extremely, extremely sad to run a microbiome trial and to not collect microbiome. And I think that it has to be part of our to-do list. And now, not a single pharma will accept to run a phase 1 trial without getting 2-3 biopsy. And, we all agree, and patients all agree for rebiopsy. So now we have to be committed. And the patient, trust me, will agree. 3 years ago, my nurse presented at ESMO that 90% of non-small cell lung cancer patients were eager and brought back their samples to the clinic. So it's not the patient, the limiting factor here. I think it's us. It's oncologists. So we have to collect microbiome. So we have to now, it's a new thing to collect. For the patient, it's easier. I would prefer to give a poo specimen than to get a rebiopsy for a clinical trial. And then you have to use this, I have to agree, a standardized method that is very easy. I think a tube that is shipped by mail to one center with RNA later, 1 or 2 tube is probably the easiest way. But please collect at least 1, if ideally 2, or not ideally 10, but minimum 2, 1 before and 1 at a month or 2 months. I will let you decide. But if you have, that's the minimum I would recommend.
I maybe echo that as well. I was really surprised when we had this discussion and I check it. We were very excited about the microbiome, but we're very concerned that it will affect accrual. And should we really go in?
No, and I can tell you the number of email I receive by patient. How can, I fax you my feces?
And that's exactly what
it changed because there's company who can do it for, a few $1,000, and then now I receive a report from company and Dr. Oti, do you know if I'm going to respond to IO? And I said, it's difficult to do. So,
and a big shout out to the patients. They were the ones telling us, no, please do that. We don't see a patient barrier. And so now we have IRB approval, so we can report back. Do we see our strong accrual that otherwise also has quite intensive blood and PRO? Will it affect or not? So I would second that, have 2 time points, or at least 1, but ideally 2 to have that baseline and then a follow-up do those early changes help you further predict your drug efficacy, I think would be quite remarkable as you're having this microbiome hypothesis to not have that translational piece. Of course, your efficacy trial is most important to get that efficacy data and to get trial results and drug results for patients. So of course you have to, maybe in a subcohort, that might be another approach, to have maybe a small subgroup where you, in patients that might be interested in just providing the microbiome, so have a very committed subcohort to do that.
I would have one thing, challenge. Think about another sample collection, which is event-based. A lot of these patients get toxicities, and we don't know at baseline if we see enough signals, or perhaps to correlate. So what we're starting to do as well is patients on immune checkpoint and radiotherapy or chemo, we add an event-based collection. That all translates to, you have to have a top clinical team, top coordination, a lot of patient communication, a lot of training behind all of that to make sure that you don't miss that sample. So
yeah, the event-based for toxicity is a real tricky business. So that I would not add to a clinical trial that is trying to get efficacy, because that is, it's such an acute setting. It needs to be a very committed team doing that acute toxicity. Those are difficult to get. And then it's also difficult to get a control sample in patients that don't have toxicity around that same time. So those samples,
I know
that's more challenging.
these things are
That's
extremely
what we're doing,
difficult.
and I check it.
They are. I think that's the reality. They're extremely difficult, but we need to move the needle, right? We need to go beyond to
great.
try to do this.
The challenge there, though, really, especially with iCheckIt, is that you want to hit it sort of at the peak of toxicity, but sometimes you don't know when that is. You don't know if it's going to get, the direction things are. If you did, it's like predicting a stock price. Is it going to keep going up, or have we already peaked? And so
onset, we do onset of toxicity.
Yeah, onset is fine, but oftentimes we're trying to hit it really at the most severe point.
And also to just reiterate a point that Bertrand made, which is don't destroy the microbiome.
Yeah.
Oftentimes we collect in Omni gene tubes or DNA preservation tubes just to make it extremely easy to ship things over mail at room temperature.
Yep.
But we are all destroying the same thing that we're going to study at the end, which is the bugs. And someone had a picture of the museum in Amsterdam. I don't know who was it.
That was me.
All right, good. Imagine all that culture that took place, right? I'd say, let's not get rid of culture if we don't have to. Like, we still need to learn about these bugs as they grow in the lab. And, just like immunologists look at, go to mouse models to identify mechanisms, we have to understand the biology of these microbes as they grow.
Yeah.
So let's
great discussion. I'll get to Seth.
Now I forgot what I was going to ask. Go ahead.
No, I was just going to say the timing is perfect. Thank you for these recommendations. Obviously, we hope these move forward, and that now is exactly the time you start talking about the secondary endpoints and also correlative. So this is perfect. We're really waiting for this workshop to get those endpoints.
Keep it simple.
Yeah.
So I was listening to Mike's talk and reflecting a little bit, and I have a sort of a question for this amazing group of experts. So the tension that I remember in the TCGA project for bladder cancer is we had in the room 2 different genome data analysis centers, which is totally crazy within a project. They took the RNA-seq data. These are world-class bioinformatic computational scientists, and they came up with 2 different results. Same data, 2 different results. I see Mike nodding his head. And Rehan Akbani, I don't know if Rehan's still at MD Anderson, spent a year with these 2 groups to get us to a point where everybody agreed on the results. All right. So fast forward, CPTAC, which is the proteomics integrated multi-platform project that succeeded the TCGA, mass spec, proteomic profiling. They spent about, 5 or 6 years just working on methodology before they ever started the first project. And it strikes me, and maybe this is happening at the NCI, I don't know, it's a question for you, but it strikes me is that's kind of what we need. It sounds like at MD Anderson, that's sort of the approach that you've taken. But as a community interested in this, it strikes me that's sort of the collaborative project that we need coming from our major funding agencies
do the hard work to work out all of these things that you've been talking about over the last couple of hours. But I also agree that you can't wait for perfection. That doing trials now is I'm going to say is probably part of the learning process, as long as our patients are, are always centered and derive the benefit of that. But it strikes me that there's I won't say a shit ton of work to get done, but a lot of work and a lot of hard work to get done, so that we learn from all this. And Mike, maybe if you can reflect on that.
Yeah, as much as I, believe in striving for perfection, the reality is it's never achievable. And so, we've talked today both about CellD. I mean, CellD is a bad technology that should have and did die, rightfully so. But, we got a lot of good information, not from CellD, but from oligo microarrays, right? And cDNA microarrays. And that data now we look back and it's pretty junky, but we were able to learn a lot from there. And so I think we're sort of in the similar stage now where we're just continuing to learn. Techniques are getting better. As for consortia that are working to understand all of these procedures, these do exist. The problem is that a lot of these studies have sort of been underfunded, and so they've not been as comprehensive as they could have been. One is the Microbiome MBQC project, and they did find similar results where different labs processing the exact same specimens had very different findings. A lot of that was driven by the extraction protocols, as I recall. So, there is still work to be done. They wish that they'd been able to do more, but it was just a lack of funding. So maybe you can convince somebody to throw, again, more Benjamins at it. It's what it takes.
Rose, did you have a comment?
I think we talked a little bit about sample collection and trying to look at data. And I think when it comes to big data, so I was born in the genomics era. So my lab started out with, when I was in training, I was working on GWAS and more to like exome, then whole genome, and now we're like single cell level, right? And so really familiar with like convoluted heterogeneous data situations. And then when I started looking at microbiome data, I was like, oh, this is a whole new level of convoluted multidimensional data, right? And so one of the things I think is really, really important if people are thinking about designing studies is that even though you want to make it pragmatic and you want to make it feasible and you want to make it easy for patients, if you can collect serial specimens, it's incredibly important because people start at incredibly different baselines. And so if you're looking at an intervention, having a before and after, or especially many time points, the more you can collect, the deeper you can go for each individual, the better you can see the change over time and how the interventions or toxicity change. And so although even in smaller studies, we're not going to be powered to detect signals in any given species or genus, or even looking at just diversity indices. I mean, these are highly variable. And so if you can get some information over time course, then you can compare with an individual. That is really, really, really beneficial. And then the other thing I want to say is I also had the same concern that patients wouldn't want to participate in my study because I was already putting them through a lot for them to bring back stool samples. And so I actually changed the study midway to have a checkbox for whether they want to bring back stool samples or not. And actually, so far, everybody's been happy to bring back stool samples to my chagrin. And then also the other thing was I thought patients would either not want to, or, so I tried to design the study in such a way that each of the collections matched with the time point the patient should come in. So I tried to minimize the number of extraneous visits. And then a lot of microbiome studies, I think, have been historically having patients go and drop them off in FedEx or UPS. That's another added barrier for patients, right? They have like insane number of things to do as it is. And so I tried to make all the time points, write them in the trial so that the patient's like, okay, I'm going to see them. I'm going to see Dr. Lee tomorrow. I will bring the sample back then because she instructed me to do so. And it's easy for them rather than, oh, I got to drop off at FedEx. And then we're calling them like, did you go to FedEx? Right. So.
And that's exactly how I checked it approach as well. It was all part of always associated with the visit. So we harmonized the various time, intervals for our cycles, the various IO cycles. So we allowed it actually 1 month plus minus 2 weeks gets us a 1 month time point, but for the 2-week regimens, that second cycle is just 2 weeks after the first cycle, and for the 1 month and then in 6 weeks, it's, so that 1 month has really a fudge factor of 2 weeks plus minus, to really exactly accommodate, to make this easy, to make it all associated with patient visits. And it cannot stress enough. So I come also from the genomics area. We were the first institution at Duke that ran genomics guided trials with RNA expression data. And it went exactly through the same method issues, Mike, that you brought up. And it was early days. So they had to still do some. It was actually a phenomenal big team that was involved with methodologists and trying to get it tumor heterogeneity, sample quality, timing, how long from the surgery was the specimen and all these aspects. But it is tough to get perfection into that. So I think we need to keep that conversation going. And I think especially within SWOG that we think we have like the best processes in place that are currently reasonably available and make sense to it. And then maybe the last comment about that baseline. So it was also my K award was about, or pre-K even, about finding baseline blood biomarkers for toxicities. And especially blood is so variable, but even tumor, it is very difficult to find baseline risk factors for future events because the patient heterogeneity is so large. You need really humongous sample sizes to really get at that. But the change over time to see the effect of therapy is a much, so each patient becomes their own control. And those early changes, I think will be particularly informative in understanding also the impact of therapy.
Dr. Gentile.
Thank you.
Oh, sorry.
Can I just
sure,
make one more comment here? Actually, 2 comments. First, the 10 years ago, 15 years ago, when I first started doing microbiome, not all patients were willing to put poop in their freezer. A lot of grandmas said, literally, we were asking them to stick it in their freezer at home. And a lot of folks were really uncomfortable with that. But that has changed. It's actually very interesting. I'm going to, for a second, take off my microbiome hat and put on my SWOG statistician hat as well and say, I agree as a scientist, it is extremely exciting to collect as much as you can. But bear in mind that as we're developing these studies within SWOG, there is a cost, not just to the patient, but also to those of us on the back end and to the development of the study as you're going along. You're putting a lot of burden on clinical research associates and so on and so forth. And the more that you load up a trial, the longer it will take to open and the more places there are for it to go completely wrong as well. And so ultimately in SWOG, the sanctity of the primary endpoint and the primary analysis, must be preserved. And so, yes, add these things in if you think it really matters, but there has to be good rationale for doing it. And at the same time, pick the time points that really matter, all right, where you really think it makes a difference, all right, whether that's at the onset of toxicity or at baseline or a month in. But, frequent sample collection, that's ideal. That works very well at single institution studies. But here, when we're doing it at this scale, it's extremely challenging.
Karen, can you comment on that? Because I imagine like in the thoracic group, as you guys were developing lung pragmatica, there must have been a lot of people saying, well, we really need to have at least one scan, or we need to have their driver mutational data and so forth. And how did you deal with that?
You're right. In lung cancer, we need to have oncogenic driver mutation information that you can't go without. And ideally, and actually we're dealing with this with the LungMap platform too, because we're trying to, the background on that is that we use only foundation and we get the raw data. And so we can analyze everything. Now in 2023, where most people get tested outside of foundation, we're trying to open that up. But if we can't get the raw data, then obviously it's less than perfect, right? So we're always dealing with those things. We came up with a case report form where people can mark, and we have all the really important ones, and then we have them upload their NGS testing and those kinds of things. So that was something we didn't want to miss out on. But like things like PROs and scan, like even some of these trials in refractory and acquired resistance, they require the scan before they start trial to prove that they actually progressed on prior IO. And all of those things we really had to push ourselves to say, no, is this going to affect the primary endpoint? And I think that's the key, is this going to affect the primary endpoint? And if the answer is no, we left it out.
And I'm just thinking
Yeah.
about the specifics of the design, because it's, sort of a targeted therapy IO versus chemotherapy trial. And what about things like TMB or other elements? I mean, would those be housed in those genomics reports
So
hopefully? Or
yeah, I mean, so we will collect that if we
Okay.
can, but we're not going to be doing correlates on this,
Yeah.
not like we are in other studies. So there are lots of other studies in the lung group in SWOG that will collect that data.
Mhm.
That's not the purpose of this trial.
Yeah, that's interesting.
Yeah.
I want to get back to that in just a second. I know Dr. Guntur has been waiting for a while here.
Thank you for bringing this rich discussion. One comment, with iCheckIt, we're doing OmniGene and OmniGut. That's what we're collecting, and Nationwide is the one that is going to go to the samples. And to answer your question regarding the events, so we're collecting microbiome at baseline, at 4 weeks, and at grade 3 or higher toxicities.
It's helpful. I actually had a question, not for anybody here at the podium, but in fact for Lucky. And so, Dr. Lara is the, newly appointed SWOG co-chair along with Dr. Hirschman, which is super exciting.
Not yet.
Not yet. It is. When is that happening, Lucky? Is that pretty soon or is that tonight? Okay. Tonight. They made that announcement a little early. Sorry, guys. Count the votes first. But I just wanted to ask you. So, here's the dilemma that we have, right? We have potential trial design like Karen's, where we sort of shed all opportunities for correlatives and so forth, and maybe just look at the important endpoint to all of us, which is OS, right? In a trial of Cabo Nivo with this live bacterial product. But then we have Bertrand and Nadim over there who are saying, no, load it up with correlatives, not just one time point, get 2 samples at multiple time points. What do we do with a trial like this within SWOG? What's the right answer here?
So the right answer is a balanced portfolio of studies that include pragmatic trials, such as Pragmatica Lung, and the trials where we load up on our TM, our microbiome studies, all of those. There's a place for everything. In the Pragmatica lung study, the goal is to identify whether the investigational arm prolongs life. Is the patient alive or dead at a certain point in time because of the intervention? All of the other things that we want to add up to that doesn't address that question. And so I think we have to now step back and think, what does a cooperative group like SWOG need to do to advance the field? And so we need a balanced portfolio. And this will always be, I think, a big chunk of our portfolio, doing studies that are pragmatic in nature, that address the real world. I was just reflecting on what Karen had said. When you're requiring a patient to have a progression scan before they enter a study, fine. But in the real world, an oncologist would make that determination, right? And so we're acknowledging what's happening in the clinic where a patient is interacting with their oncologist. And so I think that's the answer to the question. There's going to be a place for everything we want to do. And there'll be a place for all of the PROs and all the TM studies that we'd like to do.
And can I just add one more comment on that, is that we opened the study, so we got the results and presented them and published them at ASCO, and the study opened by March. Okay, so, but in order to open a study like that, as Mike was alluding to, we had to take everything out, right? And because the more things you add in and the more complicated and the more funding that you need. And so the fact that we have over 15 accruals within the first month and a half, and all of that is because of the way this is designed. And so you can't have it always for all trials.
And is there room for what I'll call maybe like a kind of pragmatica, which is where like you add in like one stool sample or something at base? I
think there could be, but I think you have to plan it very, very specifically so that you're getting the most, that it's not just undue burden, but that it's actually valuable data, right?
In Monty, essentially, iCheckIt is a pseudo pragmatica. It's a highly pragmatic trial. We started actually much more complex. We wanted to have every 3 months blood, true longitudinal blood collection at least. And the team rightfully pushed back and said, what does it add? Yes, it adds beautiful biology, and we would understand even better over time how these toxicities develop and how the biology does changes, predisposed toxicity. But we realized really the most important for predictor for the outcome for our goal is to predict toxicities, early data, especially baseline, as well as, again, that early change or that early first cycle period, how that might help us inform to really become good in predicting toxicity. So again, we scaled back many other things. And so we have this very parsimonious translational aspect to it. So baseline 1 month. And we did get that IRE time point as well. It was another discussion. Would we do that or not? Full disclosure, we don't have a control actually for those IRE time points. So we don't have that patient. That was one of, and again, maybe Mike and I should have even more conversation about that. Maybe again, a subgroup of patients potentially add that on, because we'll not know the change from baseline to IRE. Is that really truly all due to the immune-related adverse events, or is it just due to receiving checkpoint inhibitor over time? So it is this discussion of, what's really necessary for your question.
We have a mirror.
Yeah, so continuing along the theme of when to collect and what to collect. So we talked about having to look at the circulating microenvironment. I count at least 4 reasons. We talked about potential secondary endpoint correlatives, maybe achieving surrogacy at some point, looking at the mechanism by which the microbiome exerts its effect, and we may be able to surmount some of the pre-analytical confounders of collecting, microbiome specimens. So with that in mind, should we be collecting blood at the very least at every time point when we collect microbiome specimens?
For what specifically, Amir?
For all those reasons, I mean, we're going to need to be able to correlate what we see in the microbiome with what we see in the circulating microenvironment. So metabolomic markers, proteomic markers, circulating, tumor DNA, et cetera. Otherwise, how do we ultimately advance our understanding? Again, secondary biomarkers, surrogacy, perhaps, understanding of how this actually exerts its effect on the immune system and onto tumors. I think we have to collect these in parallel. Otherwise, I don't know how we would ever sort of move the needle on our understanding of these things. I don't know what your thoughts are on that and how you're designing your trials accordingly.
I think it's a good point. But as we said, the more and more we add on, the more and more complex it gets, right? So I think when we're trying to, if it's key to endpoint, right, then we got to do a smaller number of patients and maybe get more specimens in that trial. And then as you move forward, you can start shedding. So we're at the point now where it's complicated and we're trying to collect everything, but we just have to be careful about the analysis piece.
And also just the collection. I mean, again, there's a huge burden on the study itself. And so you don't collect stuff just to collect stuff, right? I mean, there has to be something going on. And at some point, no single study can answer everything. And if you try to answer too much, sometimes your study fails because you've overloaded it with so many things that you can't even accrue the patients for another 10 years. So, there's always challenges there.
Janet, thanks.
Hi, Janet Kukreja. I'm working on the transitional part of the S1600 immunonutrition trial. We kind of snuck in, so they were having like a little bit slower accrual and kind of added it on. And we're collecting at 3 time points. So before they start their immunonutrition drink, after they finish right before surgery, and then their first bowel movement at home. It's going to be super interesting to see kind of what the range of stuff we get, especially at that 3rd time point. I do want to say, like, I would highly encourage you to talk to the patient advocates about like the feasibility of these collections and stuff like that. A couple of patient advocates joined some of our calls and were super helpful and like, yes, we should do this. Yes, that's feasible. And we try to make it as pragmatic as possible in that they could mail it back, or they could also bring it back when they come to see their physician. Like there was not like one thing that they have to do. So hopefully we'll be getting our data soon.
There's a couple online questions. One was from Shilpa a while ago for Camu, which company's product was being used?
Bertrand, you want to take that one, bud? Bertrand's actually been really helpful at our site in terms of sorting this out.
So here the problem is that the active polyphenol that I presented is castellagin, and castellagin is one of the component of Camu. And so we are actually, and we just got a grant 2 weeks ago to produce capsules of pure castellagin, but this will take time, and then we have to go through Health Canada to start the trial. So as pragmatic as we want it to be, we used, and that's what we're going to do, the Health Canada approved Brazilian berry Camu Camu. And then we purchased different lots from different company. And with HPLC, we then also realized that the concentration of Castalagin was very variable. And the problem is even the same company, the lots, the concentration change over time. So what we've done for the trial is we bought 2 different lots from one company and we tested every lot, and we took the one with the highest concentration of Castalagin. So again, here, it's not the company that matters, it's the concentration of Castalagin. Hopefully, in 1 or 2 years, we will be able to provide to the patients pure Castalagin capsules.
Great. And then another online question from Austin Kirshner was April 26, 2023, US FDA approved Voust, the first fecal microbiota product that is taken orally. Any ideas about using this agent as an easy method to provide fecal microbiota transplant?
So I think this is the Vedanta Biosciences product that was just approved for C. diff colitis. I mean, Bertrand, you'd be a good person to answer.
So Vedanta has a product for C. diff. Vedanta has launched several trials in IO, and we still wait to get the outcome of the trials. Evelo has trials. We still wait for the outcome of the trials. Who else has Everimmune has trials where they just started. So CRES has trials, and they were presented at the IO trial of CRES was presented and was negative, unfortunately. So it's a moving field, and yeah.
Great, thanks. We'll close it out. I mean, I think by the number of people that were here and online, there's definitely a strong field and interest in this. And thank you all for showing up. Thanks for the panelists and our presenters today, and thank you all for participating.
Just one last comment. If you get a chance, if you see Joe Horn, I should have done this earlier. She and the Hope Foundation were the sponsors for this symposium. And also, when you look through your book, there is a QR code in there. If you want to help contribute to the science of the Vogelzang legacy, just take a picture and give them your credit card. It goes to Hope Foundation. Thanks.
Thank you.
Thanks to our panel, please.
Nice to meet you. Great. Love it. Great class. Great insights. Thank you.